Afternoon, everyone. Um, Good afternoon, everyone. It's 5 minutes past 1, and I think everybody that was waiting have been able to join. The latecomers will just miss my welcome, Biggie. So I am Anita Oberholzer. I'm the extension specialist in Enology, based in the Department of Viticulture Enology. And myself with Karen Block and Caroline Furman put it together this RAVE. It's a biannual program. It stands for Recent Advances in Viticulture and Enology. The idea is to every 2 years give you an opportunity to hear about the latest research happening in viticulture and enology, specifically at UC Davis, but not always limited to UC Davis. Although this year's program is pretty much us, everybody wanted to share what they've been doing in the last 2 years. We had no problem filling the program this year. So we want to welcome all of you. It's really sad that we had to do it virtually this year, but we rather wanted to do virtual than not at all. And we're really, really positive that we will resume in-person workshops and seminars later this year, late summer or later, but somewhere this year, we're going to see all of you in person. That's my wish. So also, I want to thank our extension partners. You'll see the screen shared with you. Without our extension partners, we can't do any of these programs because my salary is the only one that gets paid by the state of California. On the list of 3 people that I told you actually help us put these programs together. I think, one housekeeping item. If you'll see at the bottom of your Zoom, you'll see there's a Q&A button, and then there's also a chat button. Now, the chat button is for you to say hi to somebody specifically or to everybody. However, if you want to ask questions to the presenters, please use the Q&A button. If they're not able to answer your question live, because most of us like the sound of our own voice and go over our time, and then there's no time for questions, I'm going to be one of them today. I'm thinking, I hope not. But then please go into the Q&A, type your question, and the presenters will answer the question. When they're done, they're going to hang on for a while and answer your questions. And if we don't get to all your questions, we can export them and actually get them answered later to you by email. However, if you put it in the chat, we can't copy from the chat to the Q&A, so it's a big schlep. I'm just going to ask you, please put that in the QA. So please use the Q&A. I think that is everything I was supposed to say, and whatever I've forgotten, I will cover when I say goodbye to you at the end of our 2-ish hour program. So in the first session is going to be moderated by Karen Block, our industry relations manager, or director, I should say. See, my first mistake of the day. So I'm going to put my camera off and I'll see you later. Thank you.
Well, Anita will be joining us shortly, but first we have Megan Bartlett. She's an assistant professor in the Department of Viticulture and Enology here at UC Davis, and she's going to be talking about leveraging cell structure and biochemistry to make grapevines more resilient to water stress. Take it away, Megan.
Thanks so much, Karen. Thank you all so much for coming today. The last RAVE I went to was a month before I started my position here. So I'm very excited to show you what I've been up to for the past couple of years and some of the major projects going on in the lab. So today, I'm sorry, there's a chat function. I'm just going to check and make sure this is, okay, sorry. Just wanted to make sure this wasn't a note of you can't hear me or see me. Okay. So I'm going to go ahead and get started talking about projects that have been going on in the lab to try to develop grape cultivars that are more resilient to water stress. So my background is that I'm a plant physiologist and I work on stress tolerance and understanding how to use management or breeding tools to generate plants that are more resistant to different kinds of environmental stresses. So California is an ecosystem that naturally experiences really hot, dry periods. This was somewhat overshadowed by the fires last year, but the heat wave that occurred right before and during some of the fires was one of the worst heat waves in California over the last century. And we all experienced the ramifications of the mega drought around 2015, where we actually saw yields decline statewide in grapes, especially in areas that are really dependent on very high yield to be profitable. So we expect that these issues with heat and drought stress will continue to be problems in the California climate. And we also expect that climate change could potentially exacerbate these stresses. So just for example, the number of days that reach over 95 degrees, which is considered to be kind of a critical threshold for heat in grapevines, are expected to increase by 1 to 3 weeks over the next 20 years in California, depending on what region you're in. So those two things point to the fact that we really need strategies to increase heat and drought tolerance in grapevines. Especially at the same time, we're expecting to have increased competition for water coming from urban areas in California. And also some regions of California are facing stronger restrictions on groundwater and irrigation use. So one of the ways that research can help with these issues is that we can identify characteristics of grapevines that would improve their ability to continue functioning with less water, which to improve their drought tolerance, and also to improve their water use efficiency or reduce the amount of irrigation that's required to achieve our targets for yield and quality. So that's the research I'm going to be talking to you about today. So the research I'm going to be specifically focused on today is thinking about how grapevines use their stomata to regulate their water loss and their carbon gain, and how we can leverage traits that are related to the stomata to improve that water use efficiency and drought tolerance. So the stomata are these pores on the surface of the leaf that kind of act as the gateway between water loss and carbon gain for a grapevine. One of the fundamental tragedies of being a plant is that in order to gain CO2 to create carbon and sugars, the grapevines have to open their stomata, and at the same time, they lose a lot of water to evaporation. So plants actually lose about 400 times as much water to evaporation as they gain in CO2. So this means that plants become water stressed very rapidly in the process of conducting photosynthesis. So one of the things plants can do to protect themselves is that they can actually close the stomata as they start to experience more water stress to prevent tissue damage. And so when we look at these responses in grapevines, we see that grapevines actually have highly diverse responses between the stomata and water stress. Where this graph is showing the relationship between stomatal conductance, which is measuring the rate of water evaporation coming out of the leaf, versus the midday leaf water potential, which is your measure of water stress. And so this is showing plants that are very well watered, where water potential is close to zero. This part of the graph is showing plants that are very water stressed. This water potential is very negative. And this graph is showing this relationship between your stomatal conductance and your water potential for about 25 different grape cultivars. And so you can see there's enormous amount of diversity in this relationship between stomatal conductance and midday leaf water potential. But one of the first projects in my lab was to actually look at these relationships and try to say which ones are most adaptive for hot, dry conditions. Can we actually look at this diversity and relate it to the climate where these different varieties are growing? To get an understanding of what stomatal responses to water stress are especially adaptive in particularly hot and dry regions. So what we did in this study is we compiled all of the data we could find in the literature that was relating different traits that are related to stomatal conductance or to different drought tolerance traits. We're going to focus on the stomata in this talk. And so we're looking at these relationships between the stomatal traits and the climate across what we eventually compiled was a set of 34 wine grape cultivars. So you can see that this sampling encompassed a really wide range of growing season climates, where this is looking at the average temperature on the left and the average vapor pressure deficit on the right. So this is combining temperature and humidity to kind of give you an index of both heat and dryness. So you can see we got a pretty extensive range across these 34 cultivars and kind of their average climate. And the way we looked at stomatal traits in this study was we plotted these relationships between the stomatal conductance and the leaf water potential. And we summarized them in two ways. So one was we calculated this variable called the Gmax, which measures the maximum stomatal conductance when the plant is not water stressed. So this is telling you about your maximum transpiration rate, your maximum photosynthetic rate. You expect a plant that has a very high Gmax to be very productive because its stomata are very open. It's gaining a lot of CO2 for photosynthesis, but it's also losing an enormous amount of water. The other trait we looked at was something called GSP50. And what that's measuring is this relationship between GS and leaf water potential, where the P50 is the water potential that causes the stomata to close halfway. So this is a water potential. So if you have a very negative GSP50, this indicates that you're able to keep your stomata at least halfway open down to very severe dry negative water potentials. So in this example, this cultivar that's shown in black has a more negative GSP50 than this one that's shown in white. So you would expect the cultivar in black to be more drought tolerant. It's doing more photosynthesis under dry conditions. It's losing more water. This is also called isohydric or anisohydric, if you're familiar with that, where you would expect the more negative GSP50 to be a more anisohydric cultivar. Oops, sorry. So we looked at these traits across all of these different cultivars, and we related them to these average climates, the average growing region climate for these different cultivars. And so what we found is that the cultivars that are growing in hot, dry regions tend to have stomatal traits that help them to save water. So this is looking at this relationship between the maximum stomatal conductance and the vapor pressure deficit across all of these different cultivars. The colors are indicating two different ways of measuring this. That's kind of a side detail for right now. So I would just focus on the general relationship between these points. And so essentially what this is showing is that the cultivars that are growing in very hot, dry regions are the ones that have a lower maximum stomatal conductance. So they have these water saving traits. That lower Gmax can compensate for the really high evaporation rate coming from the vapor pressure deficit. It's kind of compensating for that hot, dry air and saving water. And it allows the plant to kind of extend these water resources longer into the season. It's spending them out more slowly as the season goes on. The other trait we looked at is this GSP50. So this was a somewhat more ambiguous relationship. The relationship between this trait and climate really depended on how you were measuring it. But one of the takeaways is that there's some support for having these water-saving traits in hotter regions also coming from this trait, where you can see the average growing region when they're hotter, they close the stomata, at less negative water potentials. So you would consider them to be more conservative. They're closing earlier, saving water, and avoiding the risk of really negative water potentials. So this indicates that these stomatal traits are potentially really important adaptations to dealing with hotter, drier regions. But one of the issues is that actually trying to breed for these traits or manipulate these traits is really slowed down by the fact that they're very time consuming and difficult to measure. If you look across all of the grapevine literature of the past, up to 5 decades, you're only going to find this measured for less than 30 cultivars, precisely because it takes about a day to adequately measure this for each cultivar. So it's not something that's really readily applicable in breeding. So one of the things we wanted to look at was whether or not this trait is kind of related to other traits that are more amenable to this high throughput phenotyping. And so one of the things we found is that the stomatal traits are really strongly correlated with a particular leaf biochemistry trait. And that biochemistry trait is called PIO, which is the osmotic potential. And essentially what that's measuring is the total amount of solutes that are dissolved inside of the leaf. So these solutes can be things like ions, nutrients, proteins, sugars. Essentially, the more concentrated your leaf cells are, the more negative that osmotic potential is going to be. And we think of it as a negative because we think of it as a water potential, where if you have a really strongly concentrated cell, that's going to pull really strongly for water if that plant is exposed to water. So you're measuring the strength of that pull as a water potential. Where more concentrated, equals a stronger pull for water equals a more negative osmotic potential. And so when you look at these relationships, you can see that the osmotic potential is really strongly correlated with the maximum stomatal conductance. The plants that have a more concentrated leaf and a more negative osmotic potential also have a lower maximum stomatal conductance. And they also have a more negative GSP50, where they're both more water saving early in the growing season when the stomata are open to their maximum, but they're also riskier later in the season when conditions are hotter and drier and the plant is more water stressed. So they have this strategy where they slow down their water use at the beginning of the season in order to open their stomata and be more productive later in the season around veraison when the season tends to be the most water stressed. So what we saw in this relationship was that this osmotic potential is a potential control knob or lever that breeders can work with to adapt gas exchange in grapevines to drier conditions. We can change these stomatal traits by kind of working on this osmotic potential trait. So really briefly, what this osmotic potential is doing is the dissolved solutes inside the leaves bond to the water molecules, and they help to hold the water molecules inside the leaf. So that kind of creates this positive pressure of the water pushing out against the cell walls that creates really the structural support and the driving force for growth in the plant. So if you're a plant, you have more dissolved solutes, you're going to hold in more water, you're going to be better at maintaining your structure and avoiding wilting as it gets drier in your growing conditions. So this is the same force that also opens and closes the stomata, where the stomata need to have a really negative osmotic potential in order to kind of bulge open the stomata and allow for CO2 to come in or water to go out. So that explains this linkage between the osmotic potential and GSP50. One of the issues, though, is that when the plant is watered again, you have all of the cells in the leaf have a lot of water. They have a lot of this positive pressure pushing against the cells. So that can actually kind of push the stomata a little bit closed when conditions are well watered. So that's what's explaining this relationship between osmotic potential and Gmax, where your whole leaf is built really well to keep the stomata open under water stress. But it's not as good as opening the stomata when conditions are well watered. So one of the benefits of working with osmotic potential is that previous work has developed methods to really rapidly phenotype osmotic potential, where you can adequately measure a cultivar or a genotype in about an hour compared to at least a day for the stomatal conductance measurements. So this is something that you can actually start to apply at scales that are relevant to breeding. And actually in traits outside of fruit, just looking at classic agronomic crops like wheat or rice, breeding to change osmotic potential to make it more negative actually improved drought tolerance and improved yield under drought in every species that has been tested for this relationship. So there's a lot of evidence suggesting that this is a really promising way to adapt grapevines to drier conditions. But one of the issues is that this is a difficult trait to work with because it's not something where it's either there or it's not, or it's something that has a really straightforward relationship with what we're looking for. Like for example, disease resistant, something is resistant or it is not resistant. So it's easier to go through and screen for the characteristics you want. But with osmotic potential, it actually really depends a lot on what specific value you're looking at. So if you're breeding for osmotic potential, you need to actually have a quantitative target in mind that you know is going to actually improve your drought tolerance. So for example, this is looking at 3 different relationships between GS and water potential that are coming from a low, very negative, a medium, and a very high osmotic potential that's close to 0. And you can see that this very high osmotic potential creates stomata that are too sensitive. So they're immediately slamming shut when it starts to get a little bit stressed, and they're not going to be able to keep up photosynthesis and ripening at veraison. So they're going to be very inefficient because they're not producing adequate fruit. By contrast, the ones that have a really negative osmotic potential that don't close their stomata, they're very insensitive, are also going to have problems with their fruit production because they're going to allow the vine to lose tons of water. The fruit is going to become very water stressed. And it's also going to be an inefficient use of water because you're continuing to spend it even when you could save it in dry conditions. And so there's kind of an intermediate value that's the optimum, where you're getting adequate fruit production, adequate ripening, but you're not spending more water than you need to. And so in order to use osmotic potential for breeding, we need to have an idea of what these optimums are for different cultivars and under different environmental conditions. So one of the focuses of my lab has been trying to understand what are these values that we should be targeting as breeders. So that work has been led by a postdoc in my group, Ross Deans, who's also going to be talking in the Flash Talks today. So you should check out his talk. So what he's been doing has been developing a model to understand how the osmotic potential is related to gas exchange and how we can use that model to identify target values for grapevines under their future environmental conditions. So what we're assuming in this model is we're looking at how grapevines can actually manipulate their leaf biochemistry in order to change this osmotic potential over the growing season. So this is a really widespread adaptation across plants that's called osmotic adjustment. And so what we're doing with this model is we're incorporating environmental conditions like soil dryness, VPD. We're incorporating traits, including the osmotic potential. We're using a biophysical understanding of the plant physiology to predict the stomatal conductance and the photosynthesis. And then we're adding up all that photosynthesis over the growing season in order to identify the osmotic potentials both early in the season and late in the season that are maximizing this total carbon gain. So here we're not branching out into the fruit yet. We're starting with this kind of simpler system. We're trying to say what range of these osmotic potentials would you want to give you the most carbon gain as a grapevine. And so Ross's model has shown that reducing osmotic potential in dry conditions around veraison is a really promising strategy to help grapevines cope with drier conditions. So this is showing these predictions from early in the season. This is the optimal osmotic potential. These are the optimal osmotic potentials in three different scenarios for how dry the veraison period is. This is the midday water potential that your stomatal behavior is dragging you down to. So you can see that the drier conditions, the best thing you can do is do more adjustment and reach a more negative osmotic potential. That's helping you keep your stomata open and you're reaching a more negative water potential. So really briefly, we wanted to compare this to some values that we measured during the 2020 heat wave. We missed this initial period because of COVID shutdowns, but we have some grapevines where the soil water potential was close to this negative 0.5 megapascals. That's predicted here. And so you can see that the model is realistic. We're capturing a range of water potentials and osmotic potentials that's reasonable. We're not off in space, but it's very low precision that in order to actually capture what these different cultivars are doing, this indicates that the model is going to need more cultivar specific information. And so Ross's work has pointed to a few different areas where cultivar-specific information could be really important. And so one of these is that the optimal osmotic potential depends really strongly on the actual cost of the solutes that are being accumulated. And so what I mean by that is that some of the solutes that the plant puts in the leaf are very cheap. They're just nutrients that are absorbed directly from the soil and sent to the leaf. And others are very expensive, where the plant actually has to manufacture them and expend energy. So that's going to be things like proteins. And we know when we look across plants that plants accumulate an enormous diversity in different solutes. But we know absolutely, basically nothing about what this diversity is within grapevines. But when we look at the model, we see that the model predicts that plants, that the cultivars that would have more expensive solutes would be more conservative with this adjustment. So that would be this Zinfandel and Syrah here would be an example of these different scenarios are representing increasing costs. So you can see that if these were varieties that had these more expensive proteins that they're using to change osmotic potential, they would be more conservative with this adjustment. So this work is being led by a lab tech in my lab who is joining the lab this fall as a PhD student, Gabriella Sinclair. So what she's doing is looking at how these different cultivars vary in their solute chemistry. So we're looking at differences in these low, medium, and high cost solutes. And so one of our preliminary findings is that the Syrah and Zinfandel that we saw that did less adjustment are actually using less sucrose. So they're using kind of less of this medium cost solute. So we're going to see as we move on with this work, does this reflect that they're depending more on these expensive solutes? And that's why we see that they're doing less adjustment over the dry period. And so we're also working in collaboration with the Cantu lab to try to turn this osmotic potential into a tool for developing genetic markers. So Dario has created Andy Walker created a Riesling by Cabernet Sauvignon cross. And Dario has been working with this in his lab to screen across this population for genes that are related to aroma compounds. And so we were using it in a project last summer to look at variation in this osmotic potential. And you can see that across all these different genotypes, there's an enormous range of different osmotic potentials, much larger than you would expect just from the parents. And we did some preliminary work. Jerry Lin in Dario's lab did some preliminary work relating that to the genotyping in these varieties. And we didn't find any statistically significant associations between this trait and genes, but we found some that are kind of approaching this level of significance that interestingly are really related to transpiration and water use based on previous studies. So we're going to go back out. We're going to continue working on this and expand this screening across a wider range of plants to try and develop these genetic markers that we can use to breed for osmotic potential and as a result breed for the stomatal behavior that we want. So in conclusion, this work has shown that water-saving stomatal traits are potentially a really important adaptation to hot and dry growing regions. We've shown that this leaf biochemistry trait is really closely related to these stomatal traits. But it has these really complex nonlinear effects on water use efficiency and gas exchange. So Ross has shown in his work that modeling these effects can actually provide quantitative targets when we're breeding for osmotic potential. So we know what to look for when we're screening across new cultivars. We found that diversity in the biochemistry that underlies this osmotic potential can impact these targets. And so our breeding efforts should really focus on these very low cost solutes in order to kind of get the most bang for our buck. So preliminary results are suggesting that grape cultivars do have this underlying diversity in solute chemistry. And it's also pointed to some potential genetic markers for osmotic potential that would enable us to actually leverage this in breeding. And so overall, this work suggests that focusing on this leaf biochemistry is a really promising path for breeding or potentially even genetic engineering our desired stomatal responses to water stress. So I want to say thank you so much to the American Vineyard Foundation who has funded this work, to the department and the Rossi Foundation for their support, and specifically the McElroan, Cantu, Walker, and members of my lab, especially Torsten Niepfer, Jerry Lin. Erin Galarno has been extremely helpful with this chemistry work, and she's presenting a flash talk later today. I would definitely check that out. Ryan Stanfield is a postdoc in my lab who's kind of been bridging this gap between what we know about stomatal traits and drought responses to the fruit, where he's really been working on sugar export and how that's related to water stress. So I really encourage you to check out his flash talk tomorrow. So thank you so much. I'm really happy to take any questions.
All right, it looks like we have a couple here. We may be able to get maybe to the first one, and then you may have to answer the second one. The first one is, can osmotic potential be a result of water stress? Water loss that entails higher concentration of cations and higher concentration of sugars because lack of translocation, rather than an active strategy of the plant to acclimate to water stress?
Yes, so we measure osmotic potential in these samples at full hydration. So you allow the shoots to rehydrate overnight. So in this case, you're only measuring differences that are due to the solute chemistry instead of water stress. But yeah, great question.
And there's one more question. Would you expect that rootstocks could have impact on osmotic potential in the vine?
Yes, that's a great question. So I would absolutely expect there to be a feedback between your ability to take water from the ground and do photosynthesis and then use those resources to generate these solutes. So we actually have projects, I didn't have time to talk about them, that are looking at these traits in rootstocks. And we're starting some work that's specifically looking at these rootstock scion interactions this summer. So if you get in touch with me, I would love to tell you more about that work. So please just let me know.
Great. Thank you very much, Megan.
Thank you.
All right, our next speaker is someone you just saw just about 25 minutes ago, Anita Oberholster. She's the Cooperative Extension Specialist in Enology in the Department of Viticulture and Enology here at UC Davis. And she's going to be giving a research update on grape smoke exposure impact and treatment options.
Hi, everyone. I'm just moving my presentation to a screen that I can actually see because I have 3 screens here. So I thought I'll give you a research update on grape smoke exposure impact and treatment options. And unfortunately, not as much as an update as I would have liked, but this is what we know so far. Now, I know I'm preaching to the choir here, but wildfires are here to stay. And, you know, climate experts and natural resource managers generally agree wildfires will occur with greater frequency and intensity in California, like we've seen since 2017. And it does pose a significant risk to the wine grape growing regions in the state. One of these risks is that the persistent exposure to smoke can impact the quality of the grapes and the subsequent wines. John Moramalco did a very interesting economic analysis where he estimated that there was a $3.7 billion loss from 20 billion in wine wholesale revenue in 2020 alone. So that is 3.7 billion loss just in 2020, not the previous years, and it doesn't look at the knock-on effect of other related industries like tourism, hospitality, and more. This is a huge problem, and we need to find ways to handle it and live with it. So before, I always start with what is smoke taint, and then I start with what is wine taint. So wine taint is something that comes from outside and it overpowers wine, making it one-dimensional, decreasing the quality. Now, what about smoke taint? Now, the characters of smoke taint that I list here, it's not an exhaustive list. And when I say smoky, burnt, campfire, medicinal, earthy, that's what most people see. And you can also get like barbecue, liquid smoke, and a lot of bread-like characters as well, even though you do not have bread. Something to remember, people's sensitivity to smoke vary significantly. So for one person, it may be like this, but somebody that isn't that sensitive can basically perhaps just see a masking in fruity aromas. And specifically the lingering retronasal ashtray character. Now, for me, that is the most characteristic of smoke taint, because some of the other aroma characteristics, other things in wine can give you that as well. But that ashtray for me is very, very specific to smoke taint. But there's people that can't see that ashtray. They're not sensitive to it. So they have other ways of describing smoke taint, and that's something to be aware of. Now, what happens when you have a wildfire? You have a lot of vegetation burning. Now, 15 to 25% of wood consists of lignin. It's the structure there on the left. So when you have lignin burning, you get thermal degradation, and they release a significant amount of these compounds. If you look at that, you will see this is a volatile phenol. You know, a benzene ring with an OH group is a volatile phenol. This one has a methoxy group on it, so it is guaiacol. And you have a range of volatile phenols being released in the air. Now, they break down quite quickly in the atmosphere through phytooxidation and other chemical reactions. However, they do, because they're volatile, they can spread. And also, they can use things like ash and other debris in the air as carriers to move further away. And we do not know how it impacts their stability when they're absorbing on charcoal, for instance, or carbon. And this is something we really need to investigate. And is a big focus of a huge federal grant I'm doing with my colleagues at Oregon State University and Washington State University. Now, if you have your vineyard and there's berries on the vine, and there's volatile phenols in the smoke, they will very quickly absorb through the skin into the berry itself. So it's not on the outside, it's inside. And inside, they get glycosylated through glycosylation enzyme very fast, within hours. We think this is part of the defense mechanism of the plant. So that's an example, again, of guaiacol, but we have a range of, so commercial labs will, in general, analyze at least 6 free volatile phenols. Many researchers will analyze up to 10 different volatile phenols. But we know now that they can also be bound. All these free volatile phenols can be bound to 1, 2, or 3 of a range of different sugars. So now if you think about this, this is a huge amount of potential bound compounds that can be released during winemaking and during wine aging. Now, commercial labs also have a range of 6 bound volatile phenols that they analyze, and those 7,3 and 6,3 have been shown to correlate with smoke taint development in wine. But that doesn't mean they're the only ones of interest. So most researchers will analyze way more than that, and we're still trying to see if there's other key molecules that we need to be analyzed in a commercial setting as well. Something to remember, both bound and free volatile phenols is naturally present in grapes. And we know there's a varietal impact, there's a seasonal impact. There may even be a regional impact on the amount of natural volatile phenols that would be in your grape. And this is why I always go on about baseline, because it's very difficult to interpret data when grapes were exposed to smoke if I do not know what was normal, especially in that gray zone of maybe, maybe not, when there was really just a limited amount of smoke exposure. Now, what about the timing of smoke exposure? At this point in time, we're saying the fresher the smoke, the greater the risk. And it's totally anecdotal because, like I told you, volatile phenols actually break down quite quickly. But anecdotally, we found that smoke that's less than 24 hours old, so fresher than 24 hours, has the highest risk. Also, the density of smoke and smoke taint risk they're not equal. It's not the same thing. The fresher the smoke, the more density will correlate with smoke impact risk. But the older the smoke, the less that correlation would be. We all know about that study in Australia where they found that grapes are the most susceptible from variation onwards. However, we're now saying if there's a grape on the vine and it's exposed to smoke, assume it's been impacted. Because we've now had several instances, both here and in Australia, specifically in Australia, where they have smoke exposure earlier on during their growing season, where rock hard berries were impacted severely with smoke exposure. So assume you may have a problem if there's a berry on the vine. Currently, there's no known remedial action in the vineyard. Yes, there's a lot of talk about berry sprays, and a few of them have been investigated. They've shown variable impact. Sometimes they work well, and in other conditions they don't work well. And we currently do not know, is this because of application in the vineyard? Is it because they do not maintain on the vine? And the thing is, this is something that you need to apply in advance. This is not a thing that you can apply during smoke exposure. It seems that it needs to be at very least dry on the grape before smoke exposure. We think any kind of liquid can potentially increase the surface area, increase the absorption. We don't know really the mechanism of that action yet. There's a lot of work we still need to do in this. The silver lining here is no carryover of smoke-related compounds from one season to the next. So because they're mostly in the leaves and in the berries, the next year, if your vine itself wasn't damaged by heat, next year you should be good. There should be no smoke whatsoever present in your grapes or elevated compounds of smoke in your grapes, except if there is another smoke event. In 2017, when I started my research into grape smoke exposure, all the work basically have been done in Australia, or the published work have been done in Australia. And they had these recommendations for white wines, and they're pretty much still the same. You know, for white wines, gentle processing, hand picking if you can, exclude mock and rakis, because we know that the volatile phenols also absorb onto the leaves. So if you have leaves in your fermentation, that's going to also extract into your fermentation. Rakis, we're not sure. We're investigating that at the moment. Light pressing, keep your press fraction separate. We know that mechanical action, that force actually presses more of the volatile phenols out into your juice, even for red wines that's been fermented in contact with the skins. Using fruity yeast and bulk body with different winemaking tools, it adds complexity. It just helps with masking. Now, low to medium impact red grapes. Similar, use fruity yeast, and they've recommended light toasting oak with like oak chips or other oak adducts that will help lift up fruitiness and complexity, but nothing that will also give you any kind of nutty, smoky aromas. And then the other, and fruity yeast also for masking. Rosé, that's only if it makes sense to make a rosé. Also for low to medium impact only. Something to remember, even though you will get much less of the volatile phenols because they're present in the skins with a rosé, you will have a simpler matrix. So a lower level of smoke marker compounds can stand out and still be problematic. So it's just something to take into account. I know what other people have done. If they had inventory that's not impacted, they basically just press the juice off the skins and donate skins from a different fermentation of fruit that wasn't impacted, and had quite a bit of success doing that. So in 2017, one of my red watch studies at Oakville was impacted by the fires. And so we went back to Oakville and we harvested everything that was there. And I also had grapes donated from Alexander Valley and Silverado Trail. And we decided to look at all the things the Australians have been recommending over many years in different varieties, using different fermentation techniques and different analytical techniques. Let's do it all in one and see what works and doesn't work. So I did a trial with 4 different yeasts. They've looked at many more yeast. The common denominator here was EC1118 and BDX. They tried as well. Then I tried Nobili Fresh and Nobili Base just to get oak chips with neutral or oaky aromas that will uplift fruit. The tannin is a Enology tannin. They said 100% Enology tannin can help as well. The enzyme and tannin trial was something that I did for Love Fort. At that point in time, we knew very little. The idea was to use maceration enzyme, get all the color out, give it tannin to stabilize the color, press it after 3 days, and then finish fermentation. And then I also looked at different fermentation temperatures. 10, 15, 20, and 25 degrees Celsius. The 10 degrees Celsius was a cold soak. So it was for 7 days at 10 degrees, we pressed it off and then finished fermentation. What I can tell you is yeast did have an impact. Especially BDX and D80 actually helped uplift the fruit and hide the smokiness on the nose. Now, don't get too happy about that because these were yeast that didn't perform very well, especially BDX, in the Australian study. So this is clearly matrix impacted. I didn't find that the Enology tannin did much. The Nobili Fresh in this specific matrix worked the best. The enzyme tannin treatment did not work at all. Basically, anything you do to extract the color will extract all the volatile phenols as well. So that's not going to help. And for us, for anything that had 7 days skin contact, didn't matter what the fermentation temperature was, that's similar levels of volatile phenols extracted. My biggest problem here was it was masking, but after a while, it came back. For all these wines, and I will say all of them was low to medium impacted, the smoky aromas came back and that ashy character, retronasal ashy character came back after a while. So it can help, but it's not a long-term solution. So the next year in 2018, we thought, okay, so let's say if you have smoke impacted red wine, do what you can during processing, but at the end, it's like make the best wine you can, and then look at the treatment options. So we looked at different enzyme treatments. I looked at 4 different enzymes. I know the Australian Wine Research Institute have looked at 15. I found even at 2 grams per hectoliter, 4 grams per hectoliter, 6 weeks reaction time at room temperature, we found very little hydrolysis of the bound taking place. I know the Australians found a few that worked a little bit better, but it was very inconsistent. So they're not even recommending specific enzymes for treatment. For fining, I looked at activated charcoal, specifically those that the Australians found worked really well. And then molecular imprinted polymers. Now, molecular imprinted polymers is where they use the molecular imprinting technology to basically create a cavity within a polymer that has a high affinity for your template molecule. So if say your template molecule is alcohol, then there would be a cavity in the polymer that looks like guaiacol. And guaiacol and anything very similar to that would have a high affinity to be trapped into this polymer. Unfortunately, this is not currently approved for use in the USA. Then you have reverse osmosis, both standard differential filtration and spinning cone technology. Now, I had my Oakville wine from 2017. I saw that as a low impacted wine. And then in 2018, there was the Lake County fires. And I received grapes from very close to the fire and then some distance from the fire to get a high impact and a medium impact. The high impact, we kept the pressed wine separate from the free run. So we basically have 4 wines now with different levels of impact. I couldn't do all the treatments on all the wines because some of the treatments needed excessive amount of wine and I didn't have that much. But we got a good idea. So for the enzyme charcoal treatment, this was the Vasson treatment, basically the Zimmermann treatment followed by the FPS activated charcoal fining. And then we did molecular imprinted polymer, reverse osmosis, and spinning cone. What you are looking at here is the free volatile phenols, just all added together, all the free volatile phenols I'm currently screening. And I marked them at the bottom so you can see which wines. You have the high impact press on the left, moving to the free run juice, high impact, and then the medium impacted and then low impacted wine. What you'll see is all the treatments worked quite well in decreasing the amount of free volatile phenols. Molecular imprinted polymers in general for all the wine matrices worked the best, followed with reverse osmosis and activated charcoal. They were pretty similar, and then spinning cone. If you look at the total, this is now free plus bound volatile phenols. It looks a little bit less great. You'll see all the treatments still worked, but not as well. And this is what we find. Irrespective of the matrix, they can remove more free than bound. And you see there was a little bit of a change. The activated charcoal seemed to do a little bit better than reverse osmosis, but it really was matrix dependent. Something else to remember. You do not need to remove everything. Remember, Cabernet Sauvignon has a natural amount. Any grape has a natural amount. And you only need to remove enough so that you're in the natural range, because there's been no grape that hasn't been exposed to smoke that resulted in a smoke impacted wine. I've never heard of anything like that. So basically, you need to know what's natural. The problem is we're treating wines and we do not know what we're aiming for. So it's really good if you can analyze similar matrix wines from previous years and know what you're actually aiming for. That gives you a better idea than just basing everything on taste, although taste is very, very important. So basically, matrix impact. It's very important to do bench trials. Just because one technique worked well for one doesn't mean it's going to work well for another wine. Also, I did my activated charcoal treatment only at 1 gram per liter. I mean, I just couldn't go higher than that because it's just so much, it just strips the wine so much. I know the Australians went up to 3 grams per liter. Definitely do bench trials and use the minimum amount you have to use. I would say of these treatments, they all reduce phenolics, but mostly molecular imprinted polymers remove the most phenolics, followed by the activated carbon, and then reverse osmosis, and then spinning cone. However, how much phenolics they moved was not persistent, was not consistent. It depended on the wine itself. So it really was very difficult to predict which treatment would always remove the most phenolics. I still need to look at the volatile profiles that we looked at as well to see which ones remove most of the varietal aroma compounds and you know, and esters and so on, fruity characters. I quickly wanted to talk about smoke time perception because it's really, really complex. I touched upon the fact that there's a lot of human variability. And part of the human variability is that we've found that there's bacterial enzymes in your saliva that can release the bound. And we think it's that release of the bound within your mouth that really also contributes to that retronasal character. And people that don't have that enzyme, maybe the people that can't really taste that ashtray character. It's a possibility, and we need to investigate that further. Furthermore, something like spinning cone do actually remove a significant amount of alcohol. And so that also has an impact on your matrix. Now, we know alcohol can actually decrease the perception of volatile phenols because it actually inhibits the enzyme in your saliva. The same for the sugar. Anything above 3 grams per liter in your wine can inhibit that enzyme and results in less of the bound being released during tasting. We know other volatiles like fruity estery aromas can mask smoke-tainted aromas. However, things like green aromas with oxypyrogens can actually uplift it. There's a very interesting study by Mary McKay that actually looked at this from South Africa. So we did descriptive analysis on these wines that we tried the different amelioration techniques on, through descriptive analysis. And what we did is we waited 90 seconds in between each wine, and we did a 0.1% pectin rinse in between. And that seemed to work really well. We had control wines as part of our set, wines that were made from grapes not exposed to smoke at any point in time, so that we can make sure that that there's no carryover and that people are evaluating these wines correctly. Now, this is a principal component analysis of only the significant attributes, okay? So there was way more attributes actually evaluated. Now, if you look here, this is ash, that is the retronasal character, and the peatwood smoke, that's on the nose. Now, we couldn't unfortunately do a sensory analysis on the molecular imprinted polymer wines. Those wines had very high free SO2s. I still don't know how that happened. The person that added that SO2 has disappeared. So we couldn't do sensory on that. But what I do want to show you here is if you look here on the, orthogonal and opposite to the ashy peatwood smoke characters. So here's our control wine. So that worked, right? No carryover. But what you can see here is wine 3 and 4. That's the low and medium impacted wines. Wine 3 and 4, no matter what technique we use, actually did a lot to remove all the smoke impact characters, all the smoke taint related characters. Where wine 1 and 2, those are the ones that was high impact, both the free run and the press wine, all the treatments were less effective there. So definitely matrix going to have a large impact, and you need to do bench trials to find out what works. Also, I'm not showing you the PCAs due to time, but our ashy peatwood smoke characters correlated very well with the both free and bound volatile phenols that we measured. So, and a lot of them are, even when I reduced it to only the ones that's commercially being analyzed, they did correlate very well with smoke taint characteristics in the final wine. So this is the summary. There's no 100% solutions out there, but if you think your smoke impacted, do what you can during processing to limit the volatile phenol extraction. For white wines, for red wines, make the best wine you can. I personally would go as phenolic rich, color rich, flavorful as you can, because any kind of treatment is going to impact the overall quality of the wine. Unfortunately, all treatments lack specificity, and they do remove other components that contributes to quality. At this stage, enzymes in juice doesn't work because there's too much sugars there. It inhibits. In wines, well, it doesn't work that well, but there's no damage if you try it. We are concerned a little bit with beta-glycosidases, obviously, with red wines because we have color. So I would still not recommend using a beta-glycosidase on a red wine. Fining activated charcoal works, but it strips the wine, unfortunately. Reverse osmosis, standard and differential filtration, they work, but it mostly removes the free. Spinning cone works, mostly removes the free. Flash detant. So I didn't do it because I don't have enough wine. Flash detant, I've looked at industry members that have used flash detant. It seems to work really well at masking because of all that additional color and volatile, and not volatile phenols, phenolics that's extracted and adding to the body of the wine and all the fruitiness that's there. What I don't know is how long the masking lasts. If you look at the numbers, flash detant does not actually remove much volatile phenols. It removes basically no bound, very little of the free, because the conditions are actually not severe enough. In a lab, to hydrolyze all these compounds, we heat it for over an hour at 100 degrees at a pH of 1. So that's not something close to what you grape experience in flash detant. So our recommendations for the season: take a berry sample close to harvest if you have smoke exposure concerns, right? And for yourself, even if there is no smoke, I know it's really expensive, but to have baseline numbers is so critical to be able to evaluate it when you are smoke exposed and you get those numbers back and you don't know whether this is elevated or not. Also, have extra that you put in a freezer. You never know when you need to go back to it. We're still going to recommend small lot fermentations. Small lot fermentations is just more representative because it's 30 to 4 clusters. For the clusters, wine analysis are easier. It relates more to the final wine. Additionally, you can do a sensory analysis. Sensory analysis, if people are well trained, is a great analytical tool. It gives you the best idea of what that wine will look like after fermentation. It does not remove all risk because very low impact. There may be some smoke impact still developing after malolactic fermentation and later, but it reduces the risk significantly. And I think to discuss and taste the wines is a good opportunity for growers and vintners to have a real discussion about, okay, what do we do with these grapes? Sensory evaluation of small scale fermentations, like mentioned, 20 to 25%. This is what the Australians tell me. They've been training a lot of people not sensitive to smoke taint. So if you want people to evaluate your wine, an objective panel, make sure that they're sensitive to smoke taint. Make sure you have a high impact wine to test them and a non-impacted wine, because I also find people that see smoke everywhere. Rinse with water. If you rinse with water, you have to wait 2 minutes between each tasting. Otherwise, after the first smoke impacted wine, everything tastes smoke impacted. The Australians use a very simple 9-point rating. 1 rating on the nose, 1 rating by the mouth. And a panel needs to agree with what attributes is smoke taint for them and goes into that rating on the nose and rating on the mouth. I think it's a really good system. So what I did in 2020, and I was hoping to give you an update about this. Unfortunately, I'm still waiting for instrument time. I have 1 instrument of my own. It's been in maintenance for a month. And another, I have to wait my turn as other researchers need equipment too. So what did we do? So we were recommending bucket fermentations, but actually there's not been an extensive trial. Nothing. Australians only did it last season as well to see how does bucket fermentations actually relate to standard winemaking. And I did some micro fermentations too to see how that relates to bucket fermentation. So my micro fermentations are in French presses. And we said, can we speed up bucket fermentations? I know we say bucket fermentations are 5 days. I did many last season. A lot of mine did not go dry in 5 days. You want it to be dry because the sugar can hide the smokiness. It was many times 7, 8 days before it went dry. Then I still had to settle it so that it can be clear. And often it was 10 days to 14 days before I actually had something in a bottle that I could evaluate. So we tried high biomass, high nitrogen. We tried, can we just do an alcohol extraction and you can taste that, not even having to go through the fermentation. We did it like a sous vide method. I know some people are doing. Unfortunately, we're going to analyze them in 3 weeks time. We're going to have time in the instrument and we're going to analyze all these wines. And then we also just got grapes in from everywhere. And we did both bucket and research scale fermentations for 12 different varietals from 5 different regions. I hope to go back in 2021 to those regions and get baseline data. And this is my small contribution to get baseline for the industry. Also, the question of ash versus do we remove ash or don't we remove ash? So we removed ash from Torrontés and Cabernet Sauvignon. And so that was older ash by the time we actually, our smoke exposure and ash was in August. And these wines were fermented later. So that's something we need to do more. I'm going to do follow-up bench experiments where we actually make fresh ash and introduce it into the fermentations and see what happens. I will do that before the summer so that I can tell you what's happening. We will publish that widely. Impact of rachis. People are asking me, do I include rachis or not? The Australians couldn't answer me either. So for both Torrontés and Cabernet Sauvignon, Torrontés, we did whole cluster pressing. For Cabernet Sauvignon, we did whole cluster fermentations to see if it makes an impact. And, you know, we looked at activated charcoal finding. If you know that your grapes are impacted, should you rather do it in the juice stage or in the wine stage? Which one is the least detrimental to the overall quality of your wine? We revisited rosés for Cabernet Sauvignon versus different price fractions. And then we had marvelous gifts, really wonderful. A lot of grapes donated that we made a really 9 out of 9 standard for smoke impact. And we hope to use this for training as soon as we can do more in-person training to train panels to evaluate these bucket fermentations. What's next? baseline data for different varietals, right? I'm doing my little bit, and also dialing in, why do we see different amount in different varietals, right? What's the genotypic difference that's causing this, and why are some seem to be more smoke susceptible than others? Then linking atmospheric data to smoke exposure risk, seeing can we develop low cost sensors that give us a yes, no for risk, timing of exposure, absorption kinetics, translocation within the vine. All these things have never really been sorted out. There's a lot of gaps still in the knowledge here, and we hope to address all of this season by doing intentional smoking here in our vineyard, hopefully no fires. We're looking at fast diagnostic tools, things that is not as expensive and slow as our analytical tools, just to give us a yes, no, you're impacted or potential or not impacted. If you're impacted, then you'll still have to do the detailed analytics, but at least that will cut down on the number of samples that goes to lab and inundate the labs. Barrier sprays, myself continue to look at this. I know Tom Collins is doing work on this. Australia is doing work on this. Hopefully we'll find a solution soon. And then, you know, treating smoke impacted wines. We know there's treatments there. They all work in some way or form. They all lack specificity. Finding novel solutions, you know, we're looking into developing novel resins for this, novel enzymes for this, to really do a targeted approach that do not impact the overall quality of the wine. And then sensory threshold values. What's acceptable to the consumer? This is really important. We know there's a matrix impact. We may have to go, okay, Cabernet Sauvignon, Pinot Noir, these are their threshold levels in this style of Pinot Noir and this style of Pinot Noir, and different styles of Cabernet Sauvignon as well, to know, okay, what is acceptable and not acceptable, just to help us narrow that gray zone where we do not know whether the wine will be impacted or not down the line. Okay, so take home message. I know I'm over my time. Yep. Smoke doesn't equal smoke taint. Smoke taint marker analysis. I really want to say, please do free and bound, not just one or the other. Do free and bound. It already only gives us a small piece of the puzzle, but the more of the puzzle pieces we have, the more informed decisions we can have, and we can build a database of what is normal and what is impacted. No known medication vineyard, winery, limit skin contact if you can, mask with good use, oak adducts. Something to remember, as soon as your wine has seen any kind of oak, the free number means nothing. Even if you say it's neutral oak, that neutral oak still contributes an unknown amount of volatile phenols, and you can't interpret those free numbers. You can only look at the bound and nothing else. A little bit of sugar will also help. Limit the ashtray character. Emulation, like I said, helps. Do bench trials, do a limited amount you can, and perhaps blend those wines. I don't think high impacted wines, any of these treatments is good enough, but for low to medium impacted wines, it could be a real solution. Thank you for the funding I got so far. Without funding, I can do nothing. Thank you for all the students in the lab and colleagues helping me out. And there's some photos from Harvest 2020, and I'm sure I have no time to answer questions.
That is very true. You have taken up all the question time. So you can see the questions in the Q&A, Anita.
I will be typing. Don't worry, I'll be typing.
You actually answered the first question. Jesse actually wrote below that you've answered his questions, but if you want to take a look at the other ones,
I will do so.
Feel free. Thank you. So if you can stop sharing, we'll have.
I will do that. Okay.
Whoops.
Hello, Jean-Jacques.
Hello. Here we go. Gets me an opportunity of sharing my screen. Whoops.
And while you're doing that, I will start introducing you. So our next speaker is Jean-Jacques Lambert. He's a specialist in the Department of Viticulture and Enology, and he's going to be speaking today about compost application to degraded vineyard soils, impact on soil chemistry, fertility, and vine performance.
Good afternoon, everyone, and thank you for attending RAVE.
That's your presenter view. If you want to go to the other.
No, that's fine. Is it? Do you see one slide?
Two.
Okay. All right. That's better.
Perfect.
Okay.
Thank you.
So good afternoon. Again, thank you for attending RAVE this year. The paper I'm going to present was recently published in the AJV journal, and it is essentially the work of one graduate student who is very interested also in viticulture, and today has become a professor in the Polytechnic Institute in San Luis Obispo. So this is really Stuart Wilson, and I'm sort of presenting the work he did with me and with my colleague Randy Dahlgren in soil science. So this is the results of a 2-year experiment we conducted really in Lake County, and again we wanted to test really the effects of compost application and different compost application rates on the soil chemical properties, of course, also on vine performance, and finally on the characteristics really of the fruit through the juice. Okay. So our goal really in starting this study was to see if really compost addition had an impact in terms of soil fertility. But the second point was also to measure the effect of the application of different compost rates. Okay. In previous studies, which have been published by others, we know that compost application in general to many types of crops, but also to vineyards, can improve really soil quality. Again, we increase the soil organic carbon, the soil nutrients, the microbial activity as measured by soil respiration. So this was published in 2011 by others. Then we also saw increase in nitrate, oxidizable carbon, microbial biomass carbon again, and an overall increase in soil organic matter, in nutrients, and as I pointed out earlier, also in bacterial biodiversity. Often, compost addition also results in an increase in pH, microbial biomass carbon, dissolved organic carbon, and phosphorus availability. So another point we wanted to look at really, along with others before, is what is the impact on grapevine performance really. So in several studies published in the past, people have seen that, again, you had an increase in fruit yield per vine if you added, again, compost to the vineyard. Other studies, however, did not find any significant effect. And there is even a study conducted by René Morla in the Loire Valley in France, which really, in the end, concluded that, well, there was a slight reduction in vine performance. However, if you refer to other work, as that was conducted here in the North Coast by people like the excellent Glenn McGurty, today retired, you see that the transition towards, again, organic management of vineyards generally has an impact which is favorable also on fruit quality, although it does take, of course, more than 2 years to evaluate this type of results. The area we had selected, in which we had tried for quite a few years before to really approach some growers, get funding for some studies, because some of us were interested in particular in volcanic soils, is the Red Hills area, which is a smaller AVA within the larger Lake County AVA. So currently, or at least at the time of the study, there was only about 3,000 acres planted. So there are a few big properties in Red Hills. I'm not talking about Lake County in general, okay? Especially Snows Lake at the time was a big property. And of course, the vineyards belonging to Beckstoffer, who is a large California grower. So this is an area which is mountainous. There is a lot of relief, and it spans between 1,400 feet and 3,000 feet above sea level. And of course, as a result, it also receives abundant precipitation, 25 to 40 inches, depending on the places. And another characteristic, it's why it's called red hills, really. It has these soils, very red in color, due to the presence of a lot of iron oxides, of course. So the red hills, in general, are underlain by relatively recent volcanic deposits, which tend to be high really in gravel and cobble contents. The grapes grown in the red hills are generally red grapes, especially Cabernet Sauvignon, but there is also Zinfandel, as well as a number of other varieties in smaller amounts. One of the characteristics of the area is the presence of relatively recent volcanic rocks. So this is the Clear Lake volcanic field. If you look a little bit closer, you see that it has basalt. These are the oldest deposits here on the east side of Highway 29. And then as you go toward the more recent deposits, you find andesite, also rhyolitic obsidian, and finally dacite on the flanks of Mount Konocti, which erupted for the last time only 10,000 years ago. So all of these type of rocks are a little bit different from each other due to the different composition, because they form at different temperature. But pretty much most of the soils in that Red Hill area develop from one or the other of this type of soils, including this beautiful obsidian, that black glass, that has been found all the way to the Mississippi River, because the Native Americans actually traded it. You can make excellent stone tools with it. The vineyard we ended up selecting is the Snows Lake vineyard, because somebody in the management offered us a plot to conduct our observations and experiments. The soils in general in Snows Lake are fairly clay rich. They are very red, of course. They are mostly alphisols and altisols, meaning well-developed soils. Two of the series which dominate are the Aiken series and the Kolayomi series. So these soils are very red. They also have an argillic horizon below. And again, they are very much leached because of a high precipitation amount, even though they are relatively young soils, meaning they are a little bit depleted in nutrients. This is important. They also contain a lot of coarse fragments. So their overall fertility is not very high, really, which is not bad from a viticultural perspective, because you can better control vine vigor, of course. The organic carbon typically is not very high, because after removing the forest, of course, in forest soils, generally the organic layer is really close to the surface. They don't have as much organic matter than soils from the grasslands elsewhere in California. And if you want to know more about the type of soils there, we actually published a couple of papers, one in Geoderma and the other one in the American Soil Science Society of America journal. So the vineyard we selected, again, in that Snows Lake vineyard is planted with Cabernet Sauvignon on 3309 rootstock. It was planted in 2001. And it is trained using, of course, the classic VSP system. These lands have been cleared from previous vegetation, generally chaparral and forest, and also in many cases, some of the biggest stones and cobbles and so on have been removed, which led to, in places, significant modification of the upper part of the profile. And this, combined with the fact that we are on slopes and that the erosion risk, of course, is fairly important, means that soils potentially are at risk of losing their A horizon, their upper layer. Also, the fact of having worked so much on them to level the land and prepare it sometimes has led to sort of modification of the soil horizons and modification also of the fertility of the soils in local areas. The vines were drip irrigated again. And the target stress, if I may say, on the vines during the growing season is somewhere between -1.2 to -1.3 megapascal between bloom and harvest. However, the soils receive irrigation, it's available, and also through the irrigation system, a certain amount of nitrogen and phosphorus through fertigation. Otherwise, most of the practices which are in use in these vineyards are standard for all over treatments. Okay. In terms of the experimental design that we chose, or rather before the experimental design, the duration really of the study, it was a very short study. Again, it was conducted by a graduate student. There was really no specific support for the industry. And the compost was applied really in selected places in 2012. And the effects of the compost application were studied through 2012 and 2013. So there were 3 compost rates besides the control, so 11 tons, 22 tons, and 33 tons per hectare, okay? So the compost was essentially steer manure mixed with other type of elements with a carbon to nitrogen ratio of about 12 and a half, okay? So carbon to nitrogen ratio, again, is a reflection of how quickly that compost will evolve into releasing nutrients in the soil environment. 12 and a half, or the ratio of 12 is fairly high, meaning the quantity of nitrogen in relation to carbon is fairly high. Usually the CN ratio for compost is around 20, but again, it depends on the components. In this case, there was steer manure, which can decompose, of course, more readily and therefore will release a lot of its nutrient faster than a compost would have a carbon to nitrogen ratio of 20, okay? Now, the experimental design itself was, of course, a randomized complete block design with 4 blocks, okay? So control, 11 tons, 22 tons, 33 tons per acre. All of this was distributed on 16 experimental plots, again, each with 36 vines in parallel row. And only the 10 vines in the central row were actually the object of sampling and observation. This, of course, to limit the edge effects. Compost was applied in one application, single application in 2012, and no compost was applied for the second year. I put here the composition of the compost on the bottom line. You see the compost, again, is mostly ash, of course, that's what it was mixed it. The steer manure was mixed with, of course, ash and other type of deposits, and you see that it has a healthy amount of carbon, again, nitrogen, phosphorus, and so on. So here are the 3 application rates also, and what it translates into in terms of the nitrogen, phosphorus, and potassium, which is applied to each individual vines. So in terms of sampling, again, only the vines in the center of each plot are really sampled. We collected the petioles at bloom, okay? We did not collect any petioles for veraison or harvest, and these petioles were analyzed for nutrients. At the same time, at harvest time, sorry, we also harvested the fruit by hand, okay, for the whole really plot, okay, to measure the productivity. And we also sampled about a couple of clusters, about 200 berries, okay, randomly from each vine. And on the berries, on the juice obtained, we measured, of course, the total soluble solids, the pH, titratable acidity from the crushed berries, and again, we also measured, or we calculated, the cluster numbers, cluster weights, and so on, but this was only for 2013. And finally, we also, of course, measured the pruning weights to calculate the RAVE index, even if it's old-fashioned. So in terms of a sampling of the soil was sampled in the fall, in early November, in both years, after harvest, at a sampling depth of about 15 centimeters, and the sampling was done under the row, okay? Basically where the compost had been applied. So we did 3 subsamples per replicate, and again, we took the samples a little bit outside of the drip zone, but pretty close to the vine row, okay? We measured carbon, nitrogen. We also measured the phosphorus again, including the phosphorus sorption potential in these soils, which means the ability of the soils to really dissimulate or absorb phosphorus and make it unavailable. We also measure the soil pH as well as the overall cation exchange capacity and the extractable cations. Finally, the available phosphorus, the one in the soil solution or readily exchangeable, meaning the one available for plants, was measured by bicarbonate extraction. So what are the results really of this study? Again, we saw an increase in vine yields at the highest application rate, and we saw that only in the second year, even though there seemed to be some trend overall, as evidenced by these graphs. But the only significant result really was measured the second year. For the pruning weights, again, we saw an increase for both years, also at a significant level for the highest compost application rate. So that's a fairly quick response from these plants. As far as the berry weights are concerned, again, the berry weight increase at the highest compost application rate, and this is true for both years, okay? Of course, the number of clusters is determined by pruning, so that didn't change. And the cluster weight was increased, of course, as a result of the individual berries becoming a little bit more plump as a result of the compost application. So when you look now at the vine yield, pruning weight, the berry weight, so they increase, and they increase in a linear fashion, really, with the compost application rate. And this was true not only for the second year, when we expect really a response, but also already in the first year. And once again, this experiment was conducted on soils that were determined as being degraded, meaning having lost some of our native fertility and really needing some addition of compost, okay? However, when it comes now to the compost application effect on the juice chemistry, unfortunately, there was not much effect at all. But this is not really surprising. In many studies that have been published, it does take time really to see a difference when you change vineyard management, for example, when you transition from conventional to organic. Of course, this one is not transitioning, it's just compost addition. But in this case, the fact that we don't see any difference in or any changes really in the juice chemistry is not particularly surprising. Perhaps we would need a few more years of work on that to find any differences at all. Of course, for the rest, logically, we see concentration of nitrogen, phosphorus, potassium increasing really significantly. And this is true for the highest compost treatment level, okay? Not so much for the other treatment levels. Now, for the carbon, nitrogen, and CEC, we also see an increase again following the compost treatments. This is not surprising either. Of course, carbon is being added, nitrogen is being released from the decomposing organic matter. The CEC is increased by the fact that you add organic matter, and organic matter has a high CEC. And of course, pH increase is also something that has been observed elsewhere when you add compost to vineyard soils, especially in this case, since we have a pH here of 5.8 in the control. So this is very close to the boundary with a strongly acid label, which is about 5.5. So these soils are definitely acidic. And the addition of compost here really pushes the pH a little bit towards normal. In terms of exchangeable cations, of course, we also see an increase. That's normal. Again, by adding compost, not only do you add nutrients in the soil environment, but you add also, through the addition of organic matter, you enhance the ability of the soil to hold more nutrients, okay, in the soil environment. Finally, when we look at the increased phosphorus availability, it's there. So that's kind of a positive effect, especially since originally when we tried to start working in that Red Hills area in Lake County, one of the reasons we went there is that we were told that really these soils experience a problem with phosphorus supply. So that was really the original incentive to go and work in the place. Okay, so interesting also in that graph is not only the increased phosphorus availability, meaning the phosphorus in the soil solution, but also the phosphorus on the exchange sites, okay, but the one which will be readily available for plant take-up. You have also reduced soil phosphorus sorption on minerals and other places, and this is in a sense good because that phosphorus again seems to hang more in a form which will be not perhaps directly available. Little phosphorus generally is in the soil solution, but a form which will be more readily available eventually to the plant during growth. So you see, I'm not going to last as long as my dear colleague here, because here we put the ash in the soil, not on the grapes. So I'm already at the conclusion slide. So in conclusion, a single, again, dose of compost, either 22 tons or 32 tons per hectare, it would actually translate into the same value if you use pounds per acre, for those of you who really don't like the tons per acre. So that dose applied one time really to these degraded vineyard soils is resulting really obviously in an improvement in vine performance, okay, as well as soil fertility and importantly the soil chemical characteristics. As for the effect on the vine balance or juice characteristics, well, none was observed, but as I said a little bit earlier, generally when you do that type of compost application or other manipulations like changing, for example, from conventional farming to organic farming, it takes a little bit of time. There is a lag time really before you see the actual effects of that management change. Finally, a small compost addition here, the 11 ton, had no, again, significant benefit. So at this point, it's hard to conclude anything from it, except perhaps there is a threshold at some point in terms of how much you have to add from a specific site. Also, originally in the controls, we saw some pretty low nitrogen values. And It's very likely that some of the yield limitations that were observed in these soils, it's also one of the reasons where we started that study. These nitrogen or low nitrogen, basically status of these soils benefits really from compost addition, okay, over the long term. So for viticulturists anyway, as well as for environmentalists, compost addition, again, is a good way to not only sequester carbon over the long term, but also to really provide a boost for, again, the growing vines, especially if they are in soils which are either degraded like these soils or soils which are getting a little bit tired over time. Again, the compost application has a double really effect. The first one is to enhance really the carbon content of the soil, to increase the number of exchange sites, which can possibly hold nutrients. And the second, of course, benefit is to provide directly nutrients to the growing vines. Okay. And of course, I could end up talking also about the effect it may have on soil stabilization, especially in places like that with strong slope, strong seasonal precipitation, and highly erodible soils. So in conclusion, I also want to acknowledge again the Snowflake Vineyard team, the one we started with, as well as the Gallo family vineyards who purchased the property towards the end of our study, and especially Brodie McCarthy, the Gallo viticulturist who helped us with the end of our experiments. I thank you for your attention, and if you have any questions, I'm happy to answer them. Thank you.
Thank you very much, Jean-Jacques. I think in the interest of time, like we did with Anita, there are 3 questions in the Q&A. So if you could answer those, typing them,
Okay.
we are going to, oh, there's a 4th question that just came in. So if you can answer those during the break, we can take our 5-minute break. So initially we had scheduled a 10-minute break.
Okay.
But we're just going to take 5 minutes. So it's now 2:35 and we will come back at 2:40 to start the flash talks. So thank you very much, Jean-Jacques. Really appreciate your
You're
speaking
welcome.
today and answering the questions that are now in the Q&A.
Already. Thanks.
All right. Hey there, Anita. Are we almost
Hello.
ready for our?
Yes, so next is the flash dogs, and I'm the lucky person that is gonna moderate the flash dogs and time them. And yesterday, hopefully my son hasn't changed it. Yeah, he changed everything on my phone. Don't give your phone to your 7-year-old. Anyway, I found the most obnoxious alarm I could find. It's a total ripoff from Andy Walker when he did the flash dogs for ACV, is the old car horn. So for each...
Oh, go ahead. I'm going to share my screen.
Okay.
And then you can continue talking.
Sure. So for the audience, each person has 3 minutes. I'm just going to introduce them by name and say go. And when I say go, I'm going to press the timer for 3 minutes. And they will say next because we've actually already combined their slide deck. So Karen is going to advance the slides for them. Okay, so our first flash talk speaker is Lauren Marigliano, and go.
Good afternoon, everyone. My name is Lauren Marigliano, and I'm a master's student working in Dr. Khan Kurcharal's lab. My research focuses on mitigating climate change impacts on wine grapes using partial solar radiation exclusion via shade films. Next. Each of the 4 shade films transmits a different light spectrum to the vines. Some of them vary in UVA transmission, while others vary in UVB, and others vary in near infrared. You can see in the graph on the right, the orange line, which we call D5, which reduces near infrared light transmission to the vines. The shade films were applied in 2019 and are retained throughout the whole year. We collected data for both primary and secondary metabolism, particularly solar radiation exclusion, or solar radiation at the cluster level using temperature and light sensors, ET using atmometers, which can be seen in the bottom right-hand photo, and yield components. HPLC was used to collect skin anthocyanin, flavonols, and proanthocyanidin concentrations. Next. Starting with evapotranspiration, we found that the shade treatments reduced evapotranspiration compared to the uncovered control. This is likely due to reduced temperature under the shade films. The water footprint in the shaded treatments was also reduced compared to the control. However, this is solely due to water footprint being defined as irrigation plus rainwater, and the treatments do not receive rainwater while the control does. On the right, you can see at harvest with the red line that the control had the lowest concentration of skin anthocyanins compared to the shaded treatments. This is probably due to more heat degradation of anthocyanins in the exposed control. Conversely, total flavonol concentrations were highest in the control compared to the shaded treatments. And this we can attribute to possibly excluding UV light from the shaded treatments, as UV light is critical in the biosynthesis pathway for flavonols. Next. Overall, from the first season, we can conclude that partial solar radiation exclusion produces fruit with higher anthocyanins but lower flavonol concentrations at harvest compared to the uncovered control. However, despite reduced flavonol concentrations, the shade treatment remained beneath the 7% camphorol biomarker for overexposure, which you can see in the graph on the right. Also, the shaded vines had reduced evapotranspiration and lessened vine water footprint compared to the control, which has large implications for areas like California that experience frequent droughts and have high water scarcity. Lastly, D5 produced the most stable fruit and wine flavonoids due to partial near-infrared exclusion, and this is believed to be that D5 produces the appropriate balance between heat and light exposure. Next year, we are interested in the compounded water deficit from year to year, as the treatment vines do not receive winter rainfall, ultimately reducing groundwater reserves accessible to the vines. Thank you.
Wow, you had 10 seconds to go. Well done.
Thanks.
Just something I forgot to say, if any of in the audience have a question for one of the presenters in the flash talk, please use the Q&A and they will answer it by typing an answer. Thank you. So our next speaker is Raul Guerrero. Raul Guerrero. Hi, Raul. And Raul is in my own lab and I mess up his name. Okay, Raul, go.
Okay, my real name is Raul Girardello. For the last years, I worked for Anita Berholzer as a postdoc, and for the last years, we've been investigating viticulture and performance of 4 clones of Petitia, and we determined their wine characteristics as well. Next. So as we know, grapevines cultivar and their clones, they are not genetically homogeneous. So for this reason, the aim of this study is to evaluate phenotypical and enological traits of 4 clones of Petitia over multiple seasons. So next. So this study, as I say, this study is a multiple year study that started in 2019. The vineyard we'll be evaluating is located at the Robert Mondavi Institute at UC Davis, and we are evaluating 4 clones of Petitia, FPS 01, FPS 3, 4, and 5. And as you can see in the picture, the results will be labeled as different colors, so it's going to be easy to understand. The evaluations we've been doing this vineyard and these grapes are first in the grape side, we're evaluating viticultural differences between among the clones, such as yield components. Also, we're evaluating grape composition. We're trying to see how the clones are different regarding phenolic, volatile compounds, and chemical composition. In the wine side, we also did chemical evaluations in wine composition of these wines, which we include phenolic and volatile compounds as well. And also we are doing sensory analysis in these ones. So next. So here you can see some of the tiny part of the results we found so far for grape composition and yield components. In the left corner, lower corner, we can see the 4 different clones and the chemical composition. We didn't find any statistical differences regarding Brix, pH, TA, malic acid. In the center, we see like yield components such as clone cluster weight and yield, and we also didn't find statistical difference for this. In the right side, we see anthocyanins and flavonol content in the berries. So for the different clones, and then we found that clone 3 and clone 5 had a higher content of
Is it just me or did Raul freeze?
Raul froze. All right, we may have to come back to Raul.
So what's plan B?
Plan B is why don't we go on to the next?
Yeah, because his time has just completed. Okay, let's get back to Raul. I'll send him a message right now.
Okay.
Okay, so our next speaker is Ross Deans, and you may go.
Great. Hi, I'm Ross, and I'll be talking about using adjustments in leaf biochemistry to improve grape drought tolerance. Next. So when I'm talking about leaf biochemistry, what I mean is the amount of solutes inside the leaf. And this is important because it helps keep turgor in the leaf and gives that leaf rigidity. And so this works by solutes in the cell pull in water from around it using biosmosis. And this creates a pressure back, a turgor pressure back, which keeps the leaf rigid and happy. Now, when a plant experiences something like a drought period or something like that, it becomes harder for the solutes to pull water from the surrounding area. And so what you end up with is you end up with the leaf not being able to hold as much water and the leaf becomes wilty. And while that's happening, photosynthesis is generally lower, as is growth as well. Now, plants can sort of mitigate this by adjusting their biochemistry by putting more solutes into the cell, and this creates a stronger driving force to bring water into the cell and provide a pressure so that they can do normal photosynthesis and growth. Now, unfortunately, although they're now turgid and happily photosynthesizing, they're also at a higher risk of hydraulic damage because they are operating under more water stress conditions, as well as there's potential biochemical cost from having to build and maintain these extra solutes. Now, the amount of solutes inside the leaf is a really important drought tolerance trait, except until recently, there hasn't really been a way of how to predict it under different conditions. And so I've been working on how to go about predicting how leaf biochemistry should change between wet and dry periods for grape, and so look for the changes in biochemistry that will create the greatest carbon gain during those periods. Next. And so this is an example from a grape cultivar showing that optimal change from fewer solutes under less water stress and higher solutes under high water stress. And if we want, one thing that we might want to look at is what happens if we don't do any adjustments. Say if we go to just having the more solutes and being under more water stress, we see that although we do more photosynthesis, we actually end up losing in terms of overall carbon gain because of the extra cost. Whereas if we don't do as much adjustment and keep it at the lowest thing, we actually lose out both on photosynthesis and carbon gain. Next. And so we can use this to look at how it might vary for different plant hydraulic traits, environmental conditions, and biochemical costs, and start to look at how we might want to select the different traits under different conditions. And I'd like to thank the Butler Lab, the Rossi family for funding, and the Department of Viticulture and Enology. Thanks.
Thank you, Ross. Just made it. Gita, I am quickly going to give Raul a chance to complete his flash talk. Raul, camera on. Karen, back to that. Yes, see. Okay, Raul, you had 30 seconds left on my timer when you went all freezy. So you may continue where you left off.
All right, I'm sorry for freezing. I don't know what's happened. So 30 seconds, let's go next. So here are some of the results we found in the wines in 2019 for volatile compounds. I just want to highlight on the right side of the PCA that clone 3 and clone 5, they tied together with a higher positive correlation with the most of the compounds, while on the other side we see clone 1 and 4 negative correlated with majority of the compounds. So next, I would like to thank you everybody that helped in the project, is still helping, especially Bogo that supported this project for the first 2 years. So we still are doing a next season. Thank you so much.
Thank you. Okay. Thank you, Raul. So Gita, Gita Malaya is next. And okay, let me change my chain. You don't want 30 seconds. You want more than 30 seconds.
Yeah, that would be nice.
You may go.
Okay, hi everyone. Today I'll be talking about the use of redox potential as a process parameter to predict outcome of fermentations. So currently in the industry, winemakers use temperature and Brix to monitor fermentations. And with these parameters, we can see changes in fermentation and predict problems. But often when we see changes in Brix, there's a significant delay in when the problem actually starts and when it's obvious enough for us to see it and then try to correct it. So our hope is that redox data will increase this response time. Next. For the sake of time, I won't go over redox as a concept. However, it's measurable and there are commercially available probes. In this case, we used ones from Hamilton. We also used the synthetic must, which is considered a grape juice-like media with a pH of 3.25 and a yen of 208 milligrams per liter. And we also used 1 liter bioreactors for greater control over the fermentation. And then a variety of commonly used wine yeast strains that were selected based on their use in the industry or just known characteristics that they present. Next. This data shows a complete fermentation that's fairly representative. It starts at a redox level of about 300, shown on the dot or the solid line, and it drops to a low of about negative 150. And then we also see that it starts at about 20 Brix on the dotted line and then goes to completion in about a week. And at 5 Brix, we see that the redox starts to rise from around its low point. Next. I've overlaid another run with the same yeast and the same parameters just to show that the curves are reproducible, and this run also went to completion and showed similar trends. Next. This next overlay shows a run that did not go to completion, and all of these runs were at a pH of 3.25, which is fairly difficult for the yeast to ferment in, which led to some fermentations completing and others sticking for unknown reasons. And if you focus in on the first 24 hours from about 8:15 to 8:16, we can see that the Brix data doesn't change very much, but there is great variability in the redox data between the runs that did complete and then the one that did not. And we also see that the redox levels on the one that stuck rises at almost 12 Brix, which is much sooner than the one that did complete. So in the one that stuck, we see this late drop and then early rise in redox, which could indicate problems. And then from the Brix data, we probably could have predicted on day 3 or 4 that the fermentation would struggle, but had we used redox data, we would have known at least 24 hours sooner, and that could mean taking remedial approaches sooner, and that might increase the overall effectiveness of the treatment. Next. And so the data we've collected shows that redox can be indicative of outcome, and in the future, we'd like to continue to explore the possibility that it could become predictive. And the use of redox can tell winemakers there's a problem earlier than would using just Brix and temperature data alone. And we hope to eventually be able to predict whether a fermentation will fail or proceed throughout the fermentation. Thank you.
Thank you. 3 seconds to go.
Woohoo.
So next up is, oh, I'm so gonna mess up your surname. Erin Galarno. Okay, you're ready? Go.
So I'm going to talk about my postdoctoral work with Dr. Andy Walker, looking at the chemistry of fan leaf resistance. Next. So currently fan leaf resistance is controlled by a rootstock called O3916. However, the vinifera background is concerning, and thus other breeding lines have been developed. Currently, a graduate student named Andy Nayugan in the Walker lab has done extensive phenotyping of populations, including one that is a cross of Trached and O10114, looking for new rootstock lines that could allow for control of fan leaf while not having that vinifera background. However, the mechanism of understanding how fan leaf resistance and this rootstock induced resistance allows for less fruit set issues due to grapevine fan leaf virus is not well understood. And so I am investigating the chemistry during the flowering stages of the vine or of the berries to find how potential marker and potential information about how this fruit set prevention issue is being prevented with certain rootstocks. So I collected flower stages across from initials until setting, and collected, this was April through June of 2020 on Cabernet Sauvignon on this Trached 10114 block, and this will be collected again this year. Next. So far, the status of the vine is really changing with some of the phenolics overall. However, it is not changing due to rootstock. Acids are really high at the beginning of the inflorescence, but really decrease with coming onto set. Prosciadins are collecting as it's going, and quercetin glycosides stay steady once flowering starts to begin. Soluble sugars are not changing throughout this time period. However, what we really believe is likely to be occurring is changes in phytohormones, and currently we are still running data and running samples to investigate levels of abscisic acid, auxins, salicylic acid, osmonic acid, gibberellins, zeatin, and cinnamic acid. And we're hoping to find some information across in correlation with fruit set ratios that will be indicative of what is happening due to rootstock induced resistance to fan leaf virus. Next. I'd like to acknowledge the Walker Lab and also the Wallace Lab down in Parlier, California for their chemistry assistance. And I would like to acknowledge our funding of the California Grape Rootstock Commission. In addition, I'd like to mention some that I forgot to put on the slide, including the American Viticulture Foundation, the CDFA Nursery Improvement Advisory Board, and the Table Grape Commission.
Okay. Your last slide at 3 minutes. And the sound on my phone went low. Thank you, Erin. Okay, next up, we have Dave Cheng, and you may go.
Hi everyone, I'm Dave. I'm a grad student in Dr. Oberholzer's lab. And today I'm going to talk about my research project, the investigation of the impact of Pinot Noir maturity on grape and wine composition and wine style. Next. So here's my experimental design. We harvested grapes at 2 Brix levels, 23.6 and 26. When grapes were harvested, they're split into 2 halves. 1 half were fermented at the receiving Brix levels, and they're the control groups. The other half were treated with juice replacement treatment, where we replaced the juice with a common base juice in order to keep the basic chemical matrix the same. So base juice are made from grapes harvested at 22 Brix and chaptalized to 23 Brix. By comparing between the juice skin treatment between harvest, we will be able to determine the influence of the maturity of grape seed and skins on wines. Next. Oh, by the way, please ignore the water bag treatment. We don't have time to go through it. So now I'm going to briefly talk about the wine compositional data. For the wine monomeric phenols, in the first harvest, there's no concentration differences observed. In the second harvest, we can see that the juice skin treatments have higher concentration than the control groups, indicating that there's a matrix influence. When looking at the wine anthocyanins graph, in the first harvest, the concentration is more related to the ethanol content. However, this relationship does not seem carry on the second harvest, where there's no concentration differences. And this might be caused by the limited improvement of extraction beyond a certain alcohol content. Next. For the wine polymeric phenols data, here are 2 things I want to talk about. The first thing is the difference between the control group. This might be caused by the ethanol difference. And the other thing I want to talk about is the difference between the juice skin treatment. This might be caused by the degradation of cell wall during grape ripening, leading to higher extractability of polymeric phenols into wine. Next. Lastly, we're going to briefly talk about the sensory data. So the second harvest control wine are related more to hot and persistent, reflecting the higher ethanol contents. The other thing I want to talk about, I point them out in the red circles, where you can see the second harvest control wines are separated from the second harvest juice skin treatments. And this suggests that the primary metabolites, such as sugar and organic acid, can influence how secondary metabolites are present in the wine. Next. For my research, I want to thank New Zealand Grape and Wine Institute and UC Davis Viticulture Enology Department for supporting my research. Thank you.
Anita, you're muted.
I'm so sorry. Thank you, Dave. Let me just cancel. Nazadat Torres is our next speaker. You may go.
Okay, good morning. Thank you. Good afternoon, sorry. Thank you for inviting me. I'm a postdoctoral researcher at the OP Research Station at the Cultural Lab. And I'm going to present this study dealing with the effect of leaf removal and soothing on the light and temperature accumulated by the clusters and their effects on berry and wine chemistry. Next. As you know, canopy management practices are usually employed to control the sourcing balance and improve the cluster microclimate, reducing the pest pressure and leading to a better grape and wine composition. Within the canopy management practices, leaf removal, soothing, and their combination are very popular, but their effect may not be adequate under the current warming conditions. Therefore, the aim of this study was to reevaluate the common viticulture practices in warm climates with regard to the berry and wine compositions in order to achieve the desired wine quality. Next. The zone of light intensity and the thermal time received by clusters were significantly increased by canopy management treatments. However, we found that the thresholds to achieve the maximum berry anthocyanin or flavonol contents, as you can see with the pink arrows, were different for thermal time and sunlight intensity. Next. Therefore, we could see this difference on wines. So soothing treatments increase the alcoholic content, the tray table, and the volatile acidity, and decrease the pH. They also increase the tonality, the anthocyanin polymerization, and the polyphenolic index, and decrease the total antioxidant capacity of the wines. On the other hand, leaf removal, soothing, and their combination led to a stable anthocyanin profile and decreased proanthocyanidins and fragrance trios. On the other hand, the combination of leaf removal, soothing, enhanced total flavonols, and soothing increased the mean degree of proanthocyanidin polymerization. Sorry, I didn't finish. You can see the principal component analysis on the right of the slide, and soothing treatments were clearly separated from untreated and leaf removal wines. And this separation was explained by the increased alcoholic content, acidity, tonality, polyphenolic and polymerization, while the untreated and leaf removal were characterized by the increased quercetin, catechin, and total antioxidant capacity. Next. So in conclusion, we can say that the management of the canopy should take into account the warming trends in viticultural regions instead of being applied preemptively. I want to thank to all the researchers who participate in this study, and also to the fundings that we have. Thank you so much.
Thank you. Right on time. Well, that is, according to me, the last of our flash speakers, and I would like to thank all of them for being well prepared and keeping well within their time. It prevented me from having to be rude. Thank you very much. So we're going to move right on into our last two faculty presentations of today's part of RAVE. And first up, we have David Block, the chair of the department and a professor in the Department of Viticulture and Enology, as well as in chemical engineering. Thank you, Dave.
Thanks, Anita. Thanks to all of you for being here today. What I'd like to talk about today, with RAVE being our most kind of recent advances, I thought about the various topics I could talk about, and I thought it would be a good idea to bring you kind of up to date on what we're doing in the area of low-cost evapotranspiration sensors for the idea of getting down to single vine resolution irrigation. I haven't talked about this for a while, and we've been working on especially the sensor part of this project for the last couple of years now, and this is a collaboration with several faculty here, including Mason Earls and Jean-Jacques, who you heard from earlier, Mark Burns at the University of Michigan, and most of the work that I'm going to talk about today is being done by Matt Jenkins, who's a graduate student here at UC Davis in horticulture and agronomy, and Autumn Mansfeld, who's an undergraduate in the chemical engineering department. Okay, so the outline for what I'm going to talk about today, I want to talk a little bit about motivation for this research from our group. Very briefly mention current methodology for measuring ET. I think you probably know some of that. You'll hear more of it in the talk after me by Andrew McElroon. And I'm going to talk about the components of the project, and I'm going to focus in on the sensing part because that's what we've been working on the most in the last couple of years. We'll talk about the ET sensors that we're developing for high resolution sensing, meaning as high resolution as measuring the ET of every single vine in a vineyard. And then briefly end with talking about where we're going in terms of the next generation low-cost system, because what we're working on, the data I'm going to show you now, is more of a research system, but we have started working on how we might implement this in a commercially feasible way. So we'll talk about that. All right, so let's start with the motivation. Right now, We know that irrigation changes. The needs of every vine in a vineyard are not the same. That may change, the irrigation needs may change with the topography and, you know, whether you have your vineyards planted in a hill and the vines at the bottom of the hill or top of the hill. Could have to do with soil variation that's natural in a vineyard, could have to do with disease pressure or roguing. Vines are going to need different amounts of water in a vineyard. And yet currently, as you know, most vineyards are just bulk irrigated. You open the valves, you turn on a pump, and every vine gets the same amount of water. So what we'd like to do is, given that there's a lot of water can be used during irrigation in a vineyard, depending on where you are and where your vineyard is, and the fact that water resources are scarce, we want to figure out how can we use water more efficiently, increase the water use efficiency, and provide each plant with the amount of water it needs, not more or less. So that's kind of the motivation. So the goals for this project is to sense the ET down to, or plant water status, or ET down to single vine resolution, and deliver just the amount of water, just the right amount of water to each vine. And we want to be able to do this for less than $5 a vine, maybe significantly less. And that's going to be a challenge. We'll talk about that as we go through, but that would be the goal, because at that point we figure we're talking about a 10 to 20% surplus in cost for establishing a vineyard, but having a lot more information and a lot more ability to treat our vineyard as a heterogeneous vineyard the way it is, and to treat our vines differentially. We want to develop a system that's easy to install and easy to maintain. That's going to be very important for implementation. And obviously, all of this, we hope, is going to lead to increased water use efficiency. So I mentioned this already, but regardless of the shape of the vineyard or where it's located, right now, when we're irrigating a standard vineyard, we have usually one drip irrigation line that runs down the row. And we're dripping usually on both sides of a vine, some number of hours per week that we're irrigating. And it usually involves a vineyard manager who is making a call on how much irrigation is needed at any given time of year. And then for so many hours a week, turning on the pump and opening the valves and giving the same amount of water to every vine. To measure how much water is being used, there are various methods for doing that. Everything from standard methods for measuring ET on a kind of vineyard basis with crop coefficients that Larry Williams worked quite a bit on during his time here at Davis. But there are other methods that are more automated and more specific to a vineyard. One of those is the technology developed here at Davis, Tom Shapland and others, that became Thule Technologies. And this idea is measuring the actual ET from a vineyard for a specific vineyard. And it works great. One of the limitations, though, for some of the goals that we have is that it's measuring the ET for a relatively large vineyard block, usually on the order of several acres. So the question is, can we get down to a higher resolution, meaning down to more of a single vine or small numbers of vine level for irrigation, in order to sense the water status and or the evapotranspiration? There are various ways of trying to get down to the single vine level or higher resolution. One is to use methods, aerial methods for measuring things like NDVI, an example here. And here, from the height that we were measuring, we could get down to almost a single vine level NDVI. But then, of course, there's the matter of going from NDVI to evapotranspiration or water status, which is being done by some groups, but is not necessarily straightforward. So what are some other methods? One of the other methods is the methods developed by Fruition Sciences using sap flow. And this works really well. This is a picture of the apparatus. used for measuring the sap flow through a vine, which is a direct measure of evapotranspiration. That can work really well going down to single vine level, but can be prohibitively expensive to do on very large numbers of vines. So when using this kind of technique, it's more typical for people to use, to pick one vine that's representative of the vineyard or a small number of vines that's representative of the vineyard. Doing this on every vine in the vineyard would be impractical. So again, our goal is to get down to the single vine level. And so what I'm going to talk about today is one of the methods that might be possible, make it possible to do that. So in order to irrigate down to the single vine level, there's really three parts of that. One is sensing. How do we sense the single vine ET? Then there's a matter of how we get that data to a central location and make a decision on irrigation. And then there's the whole matter of how do we deliver a different amount of water to each vine. Also not a simple thing. We did some work on that a number of years ago with collaborators out in Oakville. We were irrigating 640 vines individually with a low cost, low power system. I'm not going to focus on that today, but we did do that successfully for a couple of years. What I want to focus on is our more recent work on sensing water status down to the single vine level. Okay, so how do we do this? I'm not going to go into a lot of details. We're still kind of working out some of the details, but I'll kind of give you a general idea of how we're doing this. And as we continue on with this research, we'll give you more and more details. The general idea is how can we use kind of standard biometeorological parameters, things like temperature, wind speed, relative humidity. How can we measure those parameters and combine those with physical models, first principles models, to actually calculate ET? And so the Thule Technologies kind of approach uses an energy balance. We're not really focused on that because we feel that that's been done. And other people are working in that area, but we're looking at other approaches related to other kinds of balances like mass balances and fluid flow and things like that. Okay, so the experimental system we're using, we have 4 Zinfandel vines, and Jean-Jacques Lambert helped us to get these in these pots right away. So these are full-size Zinfandel vines in basically a container with soil. They're all on, each one is individually on a load cell here. They have sensors in them. These are not inexpensive sensors that I'm going to talk about at first. These are research grade sensors, so there's hundreds of dollars a piece. But we have relative humidity. We have an anemometer to measure wind speed. We have the load cell. We have temperature sensor that's in the, combined with the relative humidity sensor. And then we're collecting all this data on a Campbell Scientific data logger here in the vineyard. And we've been doing this for a couple of years. We've been growing these vines for a couple of years now. And we have last year's data, and is pretty good data. So I'm mainly showing you data from the last season. Because we have these vines on a load cell, we can directly measure ET. So we know what the ET is. So we can actually use our other measurements and our models to see how close we are in predicting the actual ET. So these are basically a poor man's weighing lysimeter that we have. All right, so let me show you what some of the data looks like, and then I'll show you how we combined the data and analyzed it. This is what the load cell data looks like. This is smooth quite a bit, but to give you an idea, this is the load cell data over 6 days last August in the summer. Where this curve goes up, this is the weight of the vine. Where the curve goes up, that's where we watered the plant. So you can see we watered the plant about every 3 days or so. And then you can trace this down as the mass goes down, the weight goes down. That's where the cells are transpiring. So that's the slope of this line is evapotranspiration. And then you can see every so often it flattens out. That's the nighttime when the vines are not transpiring. So the evapotranspiration, the slope at this point is 0. All right. So what we're trying to do is to actually predict this slope with the other sensors, with the relative humidity, temperature, and wind speed. So here's a kind of, for the same 6-day period, what the data looks like. This data is also smooth. Here's what wind speed looks like over the course of that 6 days in meters per second. Here's the air temperature in degrees Celsius. You can see you get diurnal patterns. And then you can see the relative humidity. You can see two curves here. One of those sensors is the relative humidity inside the canopy, and the other is the relative humidity outside the canopy. So you can see how those vary, again, with very much a diurnal pattern. And you can see separation at points along here as well. Okay, so what we did was we took those 3 parameters I showed you on the last screen, and we applied them in our models of things like mass balances and fluid flow and so forth over leaves. And the purple here is the actual evapotranspiration that we're measuring with the load cell. So this is what the evapotranspiration is. Oops, sorry about that. I was moving things around in Zoom. And I have 3 models that I'm showing you here. The MB model, you can see actually the model predicts this orange curve, and you can see that the model prediction is actually pretty good. I'll show you some more data on the next slide. In the middle model, the CMT model, you can see again, the actual ET data is the purple. And this, with the CMT model for these 6 days, that data is in the green. It's not perfect yet. We'll talk about some possibilities for that, but it's pretty good, actually, considering this is ET for a single vine. And for this FEM model, similar kind of thing. You can see this one is kind of more constrained in its prediction, but still predicts somewhat well for individual vine ET. So if we take data over the course of a couple months from last year, and we just plot the amount of water used between 1 and 2 p.m. in the afternoon, we plot the predicted from those 3 variables using those different models versus the actual from the load cell. You can see that if I focus in on the MB model here in the middle, the R squared is actually pretty good. It's 0.65, obviously not perfect. If we do the same thing for all of the data points that we collected last year, interestingly, the correlation gets as high as 0.8, which is getting to the point where it's going to be very useful as a measure of ET. Not exact, but a very good measure. So one of the things that we think is going on, and one of the ways that we think that we can actually make the model better, is that in each of these models, what we're actually predicting is a flux. So that's a evapotranspiration or water loss rate per area. And we think that that area is related to some measure of leaf area. And just to give you an idea what that might look like for the mass balance, the MB model, what we're showing is we can actually calculate that area term, because from the load cell we measure ET, and from the other part we can measure that flux, so we can actually calculate the A. And when we do that over time, and this goes from May through August, you can see that in general that trend looks something like what you would expect for leaf area over a growing season. So how will we know specifically? Well, last year we started collecting image data, and this is with Mason Earls and Stavros Viticulture from BioNag Engineering. They have this autonomous robot wandering through our vineyard on a regular basis, imaging. And so we have images of the canopy of our plants all last year. We'll have it again this year. And so the goal is to go back now and use those images to calculate a leaf area and see if we can use that to make our prediction even better and to make it less dependent on the vine. That's the goal. I will just point out that we did do a few measurements of stem water potential. Sometimes people use that to decide either on when to start watering or how much to water. If we plot that stem water potential versus basically the evapotranspiration measured from the load cell, we see there's really no correlation. If we actually take our measurements of ET versus the actual, as you saw before, there's more of a correlation. So we're not sure if our measurements will be able to tell when to start measuring, but our hope is that this will be a much better way of measuring, of deciding on how much water to add, because we'll know how much each individual vine will pass, how much water each individual vine will pass through, transpire. Well, I mentioned that the sensors that we're using are expensive sensors, hundreds of dollars a piece. And obviously this isn't going to work if we have to hang those sensors in canopies. It's not going to work from a viticultural point of view, going through and pruning, any kind of mechanization, so forth. So what we've been developing with Mark Burns's group at University of Michigan is very small and inexpensive sensors to measure relative humidity, temperature, and wind speed. And this is the first prototype that Mark developed a couple of years ago now. All of those things that I just mentioned are here on this one chip. So the sensors that we're using, the research grade sensors, are fairly large, probably about the size of a football. This sensor, all three of those sensors together are on this tiny little chip, and you can see this 3D printed cube that it fits in is about less than 3 centimeters per side. And this chip fits right into the sensor. And our goal is for the sensor, right now, these aren't incredibly cheap because they're all handmade, but we think there's a pretty good chance of getting this down below $20 per sensor. It's basically a complete weather station for less than $20. We think that not only can we get it down to 20, but we're hoping that we can get it down to significantly less than 5. The chip part of this will cost far less than $1 a piece, as opposed to hundreds of dollars for the sensors we're using now. So that's why we're very optimistic about that. This is just some data that we gathered a few years ago, and this is with Andrew McElrone's group as well. And the bottom, this is just for wind speed. The bottom is for sonic anemometer for wind speed. And what you can see is some of the different measurements from the cube at the same time. There's very good agreement. And we have a lot more data that shows very good agreement between that M cube and the measurements that we're taking with research grade sensors. I will say that this part of the project has now been funded as of last month, the M cube development by MTRAC, which is a state organization in Michigan, to try and build new technology that will turn into new companies. And so we have some money to develop the cubes, and we're developing more the software modeling side of things. So this is what a next generation system will look like. Hopefully this cube will hang on a guide wire either in the canopy or above the canopy. That's going to sense the ET. One of the next steps would be to have a control box that sits on the irrigation line that will deliver just the right amount of water. We've started working on that a little bit. Traditional solenoid valves could work from a mechanical point of view, but aren't going to work from a cost point of view. But we have some ideas on how to make a completely new class of valves that will be addressable and assignable in a vineyard, but a lot less expensive. So that's another thing that we're hoping to begin working on in earnest. All right, so to summarize, we're developing these low-cost ET sensors. The prediction of single vine ET is quite good with just the simple sensors. We're working on making it even better by including the leaf area, which you think is going to be important in these models. We have data from last year that we're going to be analyzing soon, and we have data that we're collecting this year throughout the whole growing season. The research sensors that we're using now, the goal is to get rid of them and use very inexpensive sensors. So those are pretty far along in development now. We're going to start putting these not only on our boxed vines, potted vines this year, but also in commercial vineyards where they're measuring ET in other ways. with some collaborators. So that's going to happen this year. So we'll be able to test them out. And I think with that, I'd just like to acknowledge other collaborators who worked on the project over the years, including Andrew, who you hear from next. And funding for this, a lot of the work that I showed you is funded by a gift from Till Guldeman, who I think may even be here in the audience today. So thank you, Till. In addition to funding at various times by Cypress Technologies, Cypress Semiconductor, TJ Rogers, now the funding from MTRAC and the Ernest Gow Endowed Chair in Viticulture and Enology. And so with that, I will stop sharing. I thank you for your attention.
Thank you very much, Dave. You are actually a little bit early, so you can answer. There's one question that I will in the Q&A. The low-cost ET sensor, would it be suitable for every training system?
I think we don't know enough yet to know that. So the vines that we're using are head-trained, head-proved vines. And we haven't tried them on any of the other trellis systems yet. My guess is from the data that we have so far that it's not going to matter that much. I think at some point after we get the correlation a little better, we're going to have to figure out exactly where in the canopy is the best place to put this sensor. And then it's going to be a matter of, is there a guide wire or a part of the vine that we can put this on? The goal would be to put it on the guide wire because, you know, if you go through pruning, you don't want to take these things off every season, have to put on several thousand of these per acre. Although, I don't know, if it's just resting on a guide wire, that might not be that onerous a task. But the goal would probably be to just put them on a guide wire, probably in the canopy or right at the top of the canopy. But that is something that we're going to have to do more work on to figure out the optimal siting of those, location of those in the canopy.
Okay, thank you, Dave. And with that, we're going to move to our last speaker of the day, which is Andrew McElroan, and he is a plant physiologist at USDA ARS based in the Department of Viticulture and Enology. So we like to adopt him as one of our own. Thank you, Andrew, and over to you.
Awesome. Thanks, Anita, for the introduction and for the invitation, and to all of you that organized today. And thanks to Dave for setting up my talk so effectively. He mentioned many of the things that I'll actually talk about today, and there's a lot of overlap in terms of the things that we're working on. So what I'll be talking about today are new tools for tracking water use and stress in vineyards. And it's going to scale from ground-based systems similar to what Dave was talking about, of things that would be either touching or in close proximity to the vines themselves. And then similar to the cartoon on the right-hand side, those would be on the tower systems that you see. And then actually measuring things remotely with UAV systems, both the drone copters and the bi-wing planes, and from satellites. And so I'll get into all three aspects of those as I move forward. So evapotranspiration, as you heard from Dave, has been very commonly used in irrigation management for a long time. So this incorporates the evaporation from the soil as well as the water loss from the plants themselves through the leaves. So they're opening up their stomata in order to gain carbon for photosynthesis, and they're losing water at that time in this exchange that goes on. And it really integrates very well, this ET parameter, because the plant is in contact with the water that's available in the soil. You have the roots taking that water up. You have the plant regulating that water loss by closing and opening their stomata as they're exchanging carbon. And then you have the atmosphere that's actually driving, the atmosphere and the environmental conditions that are driving that evapotranspiration of water. I really enjoy this cartoon that's from PMS Instruments, one of the pressure bomb companies that I've used several times if you've seen me talk before, because it really integrates what the plant is dealing with and what you need to think about as someone that's doing irrigation management. And it gets you thinking about the three questions that we really need for irrigation management. So how much water is needed, when to apply it, and where to apply it. And Dave started touching on the where there and several other aspects of this. But with the cartoon, you can think of, for example, with the how much water is available in the soil, how much water is being used by the vines during that time, and how much should we put back on? When you apply it is going to be related to the stress, like the cartoon images on the right-hand side of this, where you're seeing flagging of leaves and leaf loss and leaf rolling, is we want to avoid periods where we're actually driving the leaves off of these plants. But in wine grape production, we're also pushing them with deficit irrigation strategies to stress them enough to improve fruit quality and things like that. So I'll come back to these questions: how much, when, and where throughout the talk. So we also do this within a water balance concept. So ultimately what you're trying to do in your vineyard systems is balance your water budget. And this is really important for regulation that's coming online right now. So ultimately what we're trying to do is measure the inputs and outputs of the system as effectively as possible. And Dave really set this up well with needing new sensors in order to do that as precisely as possible, with very fine spatial resolution as well in the vineyards. And so you have the inputs of precipitation and irrigation into the system. You can think of this like a potted vine on your porch. So this has a plate underneath it that would normally be collecting the water. You add the water into the top. You may get some spillover from that surface. That would be the runoff, which would be affected by some slope of the vineyard or other things. You have the water loss from evapotranspiration, so ET coming out of this, and then you have drainage coming out the bottom. And what we ideally want to do is put the water where the plant needs it. So if you put too much water on, you actually saturate the soil and you get drainage out the bottom, and it carries things like nitrates with it. And that is starting to be regulated by SGMA, so the Sustainable Groundwater Management Act. So growers really need to pay attention to this, of all aspects of this, and measure it as precisely as possible to give the plant what it needs and where it needs in order to do this. So there's lots of factors that impact water use and needs. You have evaporative demand of the climate, growth stage of the vines, canopy, and trellis type, like the question that we just saw in the chat, vine row spacing, vineyard slope and aspect. Is there a cover crop there that's using water as well? Are the vines healthy? Are they having trunk diseases? And that's going to be related to their age, or viruses or other things that will impact their water use. Is there a hard pan that will impact that drainage out the bottom of the system? And where exactly are the roots? And do you need a leaching fraction? Are you in a region where you have high salinity and you need to wash those salts out of there and make sure that you're including a leaching fraction? So the way that this has been done for a while, and Dave alluded to this as well, is the seminal work of Larry Williams in our department of using the California Irrigation Management Information System, so CIMIS, which consists of a series of weather stations that are placed throughout the state on model grasses or reference plants like alfalfa that are well watered under those conditions. And so under the current climatic or weather conditions that that station is seeing in that current day, you can calculate what a plant, what this model plant would use under those conditions. And then you multiply that by a crop coefficient for, in this case, grapevines, to get to the calculated ET that we want for the irrigation. Larry actually used the weighing lysimeter. The picture here shown in the bottom left is from the Kearney Ag Center, where he calculated the crop coefficients from these two vines in this large pot embedded in the vineyard there at the weighing lysimeter vineyard, and developed these KCs that moved throughout the world. Actually, they're used very commonly throughout the world. But you can think about this in terms of, it gives you one static measure of what a well-watered grapevine would be using under those same conditions. So it's a blanket for the entire plot of land. So like Dave mentioned, we really want to get to better spatial resolution and better precision management in order to do this. The other thing about the system is that it assumes optimal conditions. So that the plants are healthy, that they have optimal nutrient and water. And the lysimeter actually used to pump water in every time 2 millimeters of water were used, it put more on. So these were very saturated vines that were actually using this, that the crop coefficients were developed from. So really what we want to get to in precision management is, how do we layer in more of these details? So to include soil type and uniformity, we know that that can be quite variable, making sure that we know about the variety in rootstocks, which can change even within a block, depending on whether you're on the slope or the valley floor in some cases. What is the phenology throughout the season? What are the age of those plants? The health and nutrition that I mentioned before. And then certainly in wine grape production, we impose water stress purposefully in order to improve fruit quality. And so how do we account for all of this in what is a sea of vines in a very effective way to actually get to these points? So the methods that I'm going to talk about today are all based on energy balance approaches, like Dave suggested, is that we actually have been using energy balance approaches to do this for a while. And I'm going to start with the ground-based measurements with the tower on the right-hand side, But all of the techniques that I'm going to mention today are based on thermal applications. So what we're doing with this is we're trying to measure the energy that's coming into the system. And that's mainly from the sun, that radiation that's coming from the sun. And then that energy gets partitioned into three components. It goes into heating the air. So some of it's absorbed by the canopy. It gets re-radiated and it heats the air, and you have energy exchange with the air as it's moving by. Some of that energy goes into heating the ground, and the remainder goes into what is latent heat flux. And this is what's actually evaporating the water from the system. Latent heat flux is very difficult to measure directly. It's expensive and very technical. And so what we do with all of these methods is we actually measure those other three components. The net radiation and the ground heat flux are relatively easy to do. The sensible heat flux is a bit more complicated with the turbulence that's involved, but that's what we aim at. And so we measure those three effectively. And then by knowing those, you can calculate the energy that's actually going into evapotranspiration, because all of the energy needs to be accounted for in the system. And so we spent a lot of time working on the sensible heat flux side of it. And this is just an example of a simulation of, it's actually real airflow moving across a crop surface. You see the air at the bottom of this is moving slowly at that boundary layer where it's impacting with the canopy itself. And then what's happening is that you're actually having ejection events. You have the wind moving from the left to the right on your screen. And as it stops there at the end, you can see these three ejection events that actually came out. So this is actually energy being exchanged with the air as it's moving by from that canopy. And that's what we're actually trying to pick up with these ground-based sensors for the energy balance approaches that we're after. And so in doing this, you have to account for that wind speed and direction of where it's coming from. And so we end up using wind roses to identify the best place to actually put the tower. And you can see here it's positioned in the center of this wind rose. And in this plot specifically, you have the wind coming from the northwest predominantly. And so we position the tower in order to pick that up. And then you would be accounting for a footprint that would be coming mostly from up in this region, but it does change at different times throughout the year. So I show this figure. If you've seen me talk before, you've seen this probably a million times, and I'll be quick with it, but it sets up what we're doing in terms of new technologies. And so Tom Chaplin was my first PhD student when I was at UC Davis. And we worked with Chalta Palu and Rick Snyder on what Chalta originally developed, surface renewal theory in the late 80s. And it's this idea that it actually comes originally from chemical engineering. And the idea is that you have this surface, and in this case, it's a crop surface. And you have these air parcels that come in contact with the surface. They settle in, and then they start to exchange energy. In this case, it's actually going from a lighter color to more red. So it's actually increasing in temperature here. And you can see that from the theoretical temperature range. So we actually measure this with these fine wire thermocouples above the canopy itself. So they'd be sitting here. And what happens over time is that the surface gets renewed. So a new air parcel comes in this third image. It bumps out the one that actually heated up. And then the temperature actually drops at that point. And this repeats over and over and over again. What Tom did in his PhD was successfully figure out the mathematics behind this of where the signatures or the signal was relative to the noise in these actual temperature ramps, and remove the need for expensive equipment in terms of this calibration. And as Dave alluded to, Thule is out there in the marketplace now providing this and doing a nice job of actually doing that on the ground. So we went from this research grade equipment to this commercial software stack that they developed, including the hardware side of it that's actually in the field. And then we calibrated it when Tom was still in the lab as a postdoc, and we're using modeled net radiation, which is what they use for the commercial system. They actually generate their radiation values from the GOES satellite, which is a geostationary satellite over California. And they actually calculate their net radiation, and they assume some things about the ground heat flux on a daily basis. But we found really good relationships between that, the ET from surface renewal with these modeling parameters and the lysimeter, and also using soil water balance approaches here, really good correlations between that. And so the method works really well. There's other companies that are now doing this. So that was almost 10 years ago that we were working on that originally. Arable has a pretty slick sensor that's out there now, and they have these arable disc sensors they use the same sort of water budget approaches and a variety of sensors in there, similar to the types of things that Dave was talking about. but more on the energy balance side of things that they do this. And so this is another example of a technique that's out there that's actually measuring this on the ground. So coming back to these questions about how much, when, and where to apply the water. So what we actually need is we need to know how much water is coming off, and that's what these ground-based sensors, the surface renewal and the airable sensors and these flux towers that we work on. But we also need to know when. So how do we measure stress? So this has been done for a long time with pressure bombs, which requires manual labor or pyrometry, and you get point measures of single leaves, and it's not really possible without a lot of manual labor to cover that much of a vineyard and understand the variability. You have soil moisture sensors, which also suffer from being point measures themselves, and are they closely located to the roots and representing what the roots are actually seeing. You have the aerial mapping, and I'll come back to this. But something that's been around for a long time is actually these infrared sensors. And since we base a lot of what we do on these thermal approaches, we wanted to actually see how the thermal sensors work, to see if we could combine both of them into a single sensor that really gives us reliable data for how much water is coming off and when it's coming off. And so we started playing around with these apogee sensors, and former postdoc Chris Perry actually did his PhD with the founder of Apogee. So we got some early sensors that were higher frequency. So we started measuring them at 1 and 4 hertz. So 4 times a second or once per second with that. And these sensors have actually been used for decades to do stress indices in plants, and they work very well for this. We actually did this in the Tyree vineyard. It uses similar energy balance approaches. And this is one of the indices, this canopies to model conductance versus what we actually measured with those leaves. And we found good relationships with a variety of these. So we felt comfortable from this and from work from Krista Shelley and other people up in Idaho that these sensors actually work for stress, but could we actually get them to the how much water is coming off? And so we started pointing them at the canopy. And this is a plot. The upper is the canopy temperature measured with the Apogee IRT sensors. And the bottom is what we were measuring above the canopy with a fine wire thermocouple at the same time. So this is the surface renewal approaches that we were doing in the past here, showing those ramps of where that energy is being exchanged. And what you can see is that you see a ramp here that's going up with the thermocouple measuring it above. And you actually see a dampened but similar ramp actually in the canopy temperature if you're measuring at 1 hertz. So there is this thermal inertia. It takes longer for that energy to exchange from the canopy itself, but you can actually see this. And what's really exciting, took us several years to do this, and a new postdoc coming into the project with better skills in biometeorology to really guide us through this, Nico Baumbach, is we submitted a paper on this just recently after lots of testing at the almond lysimeter site. This is the same lysimeter that used to contain the grapes. And we've also done it over grapevines and in alfalfa. And in the bottom right-hand plot, you can see some diurnals of what this new wavelet method is doing. So we actually, without getting into the details of the wavelet method, we use the IRT, measure it at 1 hertz, and can actually see the changes. This is the sensible heat flux that actually comes from that compared to what the surface renewal and eddy covariance is measuring on a daily basis. And then you see on the left-hand side, the correlations between what we're seeing from eddy covariance, which is essentially the gold standard for measuring sensible heat flux, and what we're getting from this wavelet method. So very good relationship here for 3 different cropping surfaces. And we're pretty excited about this because it not only gives you information about the stress and how much water is being used, but we can actually point these things specifically at the canopy of the grapevines to know what the grapevines are doing. And we can point them at the cover crop. And we can then start to partition how much water is coming off the middles versus the grapevine canopy itself. And then we can also use this as ground truthing for remote sensing technology. So all of these remote sensing technologies need some sort of waypoint on the ground to give you some information, and to be able to collect the sensible heat flux on the ground, the canopy temperatures themselves as raw values with these IRTs, and a stress index at the same time to help balance what we're doing at the remote sensing side is pretty exciting for us. So that leads me into the remote sensing side of things that we're doing. I've been involved in this GrapeX project now for several years. It actually started for a couple of years before I got involved. And this has been this really beautiful partnership where USDA, and specifically led by Bill Kustis, who is an ARS scientist working with Martha Anderson and a whole team of people at the Hydrology and Remote Sensing Lab in Beltsville, Maryland, They developed models using remote sensing data from thermal images of the Earth's surface to calculate ET using those thermal images using similar energy balance approaches. We've partnered with NASA scientists on this, Cal State Monterey Bay, Forest Mountain there, that I'll talk about a bit more later. Utah State is the Aggie Air team that we do drone work. And all of this has been possible by working with Ian J. Gallo. They actually have a former postdoc, Mimar Alcina, worked with Dave Smart for a long time in our department, and she's now working within Gallo's team and implementing remote sensing techniques within the irrigation management. And all of the other Gallo team has been really instrumental to us pulling this off. So ultimately what we're trying to do is use our ground-based sensors to truth what's going on with these ET models at a higher level. So we have towers, we have all sorts of sensors in there, and we're really refining this toolkit to make it be a functional tool for having actionable data to actually implement irrigation in real time, down to what is 30 by 30 meter resolution. So this is a vineyard in Lodi where we've been doing this for a while and have resolution down to 30 by 30 meter for doing this. The other really exciting part of this, the head that just popped up in the bottom right-hand corner of the screen, is Kyle Knipper. He was a postdoc with Bill Kustis and Martha Anderson back in Beltsville. And he just recently got hired into the new Sustainable Agricultural Water Systems unit that was just established here in Davis. And he's going to be the remote sensing scientist focused on implementing this toolkit, churning out this model data for woody perennial crops in California with a large focus as we continue to do things in grapes. So it's really exciting that he's going to be here and specifically developing the tool for California growers. So again, the same concepts are being used here. Slightly different that we use a two-source energy balance approach, or they use a two-source energy balance approach in their models to do this. And what that's accounting for is the soil evaporation, versus the vine or the tree transpiration that's coming off in the system. And you can imagine under flood or rainfall irrigation versus drip, you'll have very different situations in terms of how much that middle is wetted, if you have cover crops in there, as I mentioned before. So being able to partition the ET between what your target crop is doing and what those middles is doing is really important. And this two-source energy balance approach allows them to do this within these models. So they figured out how to take thermal images from satellites and drones and then apply this math to it to figure out where the energy is going in the system to be able to partition that into what's going into evaporating the water for the ET in these different parts. So what we do on the ground in these experiments, this is an image on the right-hand side of the two plots in Lodi where this experiment originally started. And then we really measure the hell out of everything. So we put in a whole bunch of sensors on the ground. This is an example of one of the flux towers where we're actually doing this profiling of the atmosphere above that to get at eddies of different sizes that would be contributing to this. We have soil moisture sensors arrays. There's been sap flow sensors in there. There are cameras watching or taking pictures of the canopy as it developed. We're measuring LAI and all sorts of things, including Gallo has allowed us to completely defoliate vines throughout time to actually get at those leaf area indices and things that are so important for these models. And then we also do these intensive observational periods. When the satellite is actually passing over, we are on the ground doing a whole bunch of measurements, whether it's leaf gas exchange sorts of measurements, setting up Bowen ratio systems or other flux in the middles to measure that at the same time, which can't stay there all season long. And then having the UAVs actually come over from Aggie Air to do this. So a variety of different things to partition that energy to make sure that we're measuring it as effectively as possible and to really do that ground truthing in as effective way as we can. So the Aggie Air team that I mentioned previously is currently led by Alfonso Torres and his team. And he uses a couple of different UAVs to do this. He has the quadcopter drones that are lower flying, and then he has bi-wing systems. He used to have one that needed a catapult to be launched. This one now actually hovers up and then takes off and flies at as high as 1,000 meters as he's doing some of this. And these are examples of the mapping of ET, high being the darker colors here, low being the lower, in these two plots that we've been working at in Lodi for a long time. And with his partitioning methods, using the two-source energy balance approaches, we can also know how much of the ET in this space is coming off of the vine transpiration versus the total ET from the middles in the middle of that as well. So Coming back to the idea of where this originally started with Landsat satellites, this is an image from Kyle showing the 3 different domains that we work in. So each of these is something like 2 million acres that's represented by those domains in each of those squares on the right-hand side of this. And then the vineyards that we actually work in. So these are examples from Sonoma, Lodi, and Ripperdan. We currently have 13 flux towers in a variety of different vineyards throughout California across this range. And these have been up and running and will continue to be, hopefully for the foreseeable future here, for doing these ground truthing and lots of other experiments that we're doing. But you can see that it's mapping across time. This is a data set from, from the season where you're going in 2018, from April to May, up to September, and how it's changing in those plots over time. And you can see the surrounding vegetation or native areas changing in the ET levels relative to the irrigated plots in each of these, and how much it actually goes up in different locations, and the differences in ET that we see going from the northernmost to the southernmost sites, where you'll actually have more irrigation input and much larger canopies in this lower area. This is zooming in on that Lodi site again from 2014 through 2016 of data that he collected across the field season. So the measured is what was coming from the flux towers in the red dots. The modeled is the black line of what that is for the average across that plot. And you can see that it's actually matching pretty well across that time of picking up the ET as it changes throughout those seasons. And what the gray is actually showing is the spatial range. So how much variability within these plots actually exists in terms of the ET from one location to another. And we also are doing this on canopies of different sizes. One of the reasons that this site was chosen is I had a more mature vineyard on the northern site and a younger one on the more southern part of the site, and the differences in ET that you see from this. And so this variability that you're seeing around these means really points to why we need the mapping tools in order to pick this up and deliver the water most effectively. So in the last couple of slides, I'll finish up with this, is we're also doing it in the variable rate drip irrigation system that Gallo developed with IBM and others. And we were aiming to deliver the water down to 30 by 30 meter resolution. And in the plots that I show you in the next, this is at the Ripperdan site. It's been split up where we were aiming for the most stress in the northern 2 and moderate or no stress in these bottom 2. And we tracked it over the season. And this was a system that was developed originally by Luis Sanchez with a lot of other collaborators that he was working with at Gallo. And so this is from Kyle's plots demonstrating the prescribed irrigation as we were heading towards. We had a targeted date in late July for when we were going to do an IOP. And we wanted to get to that point where we gradually increase the stress to that point. You can see pretty uniform ET throughout the entire plot. This would then again be split into the more stressed plots at the top, no stressed at the bottom, and the ET that needs to be prescribed based on each of those blocks of what's coming off of how much needs to go back into that. And then as we progress through the season here into July, as we're building up to those bigger differences in terms of those treatments, you start to see ET drop in the top, the no stress treatment, getting more water, actually holding that higher ET, and then really very dramatic differences here at the end of the season. We were really driving stress in these upper plots so much so that we burned some leaves off of those. But demonstrating how you can use this ET with prescribed irrigation to do this. This tool is actually coming online via the OpenET system. This is this incredible project that was started by the Environmental Defense Fund as really a policy project in partnership with NASA and Google Earth Engine. Forrest Melton at Cal State Monterey Bay has really been the driver of a lot of this and the coordination of all of these different groups. And what OpenET is doing is aiming to map ET across the whole Western US. So you will be able to go down to, similar to this droplet that's shown here, plot level data for getting ET within season based on these models that are there. So the models that Kyle is using, that Bill and Martha developed, are being ingested into OpenET, and they're being compared against other models. And I'll show those on the next slide. But ultimately, it came out as a policy tool originally in terms of water allocation and water use. But the agricultural community said, we want in on this. This is going to be an important tool for us that we can use to manage our water and will be really helpful. And part of the Gallo team is actually embedded in OpenET as an advisor to what they're doing. So these are the 6 models. So ours is the model that Bill and Kyle and those guys developed is the Alexi Dyslexi, but there are lots of other approaches to this. And so there are 6 that are well established and reputable, and all 6 of these are available within this OpenET platform. It was supposed to be online fully as of December 2020, and it should be within the next couple of months available for all users. So this is going to be a tool that's available for you to access in an effective way for ET data in the future. So in conclusion, I showed you some data about new ground-based sensing technology and remote sensing that gets at the amount of water. We are feeling very hopeful that we can combine those into a couple different sensors to also get at the stress side of things as well. And similar to what Dave was talking about is we need methods to actually deliver that water spatially to account for that variability that we're seeing. Not everybody has a variable rate irrigation system like Allo has, but there are companies coming along that are developing this. And WaterBit was actually doing this. TJ Rogers was involved in this company, and they recently were acquired by Farmex. And so they are working with a variety of different people to develop these irrigation systems to get this out and deliver water variably within the vineyards. And so with that, there are a whole lot of people that were involved in this work over time. And without naming all those folks, I'd like to thank them and the funding sources that actually made this all possible, and we'd be happy to take any questions if there are any.
And I am muted. Okay, thank you, Andrew. We're actually over time, but there's 2 questions in the Q&A. And because we're ending the program, I wonder, perhaps you can answer the second one quickly, typing that, because that's going to be a shorter answer. But the first one is, how does terrain variability affect these methods? So they say we used the Thule system for 1 season several years ago, and it was totally uncorrelated to reality. The conclusion was that our terrain varied too much within small areas, example, less than an acre, such that the eddy current measurements were not useful to predict ET.
So that's a really good question. As a federal employee, I will say I'm not involved in what Thule is doing anymore in terms of understanding exactly what's going on behind the curtain a little bit for that. But there is a spatial scale with which you can actually get these measurements in terms of where the wind is coming from. And if there is a lot of slope, like you're talking about in topography, you can have drainage of cool air coming down hillsides that will impact those energy balance approaches. We are doing a lot of work right now for trying to understand heat sources outside of the plot that we're actually working in, or cool air coming off of slopes like that to actually better understand what's going on within that context. But yes, that is one of the things that we have heard that the Thule system doesn't work that well for it, and one of the reasons why we're exploring these other techniques in order to continue moving this forward for ground-based sensors.
Okay, can you answer the second question in the Q&A while I say my final words? Would that be okay?
Yes, I could do that. That sounds great.
Thanks.
Thank you.
So I would like to thank everyone. I would like to thank all our speakers for making this a successful rave. We're not done. We're going to continue tomorrow morning. And I also want to thank all the attendees, because if you're not interested in what we're doing, then there's not really much point of us doing what we're doing. I would like to thank Caroline Furman. She's our program representative, and she does all the stuff that I will mess up, like registrations and Zoom links and everything, logistics. And our industry relations director, Karen Block, that is really the driving force behind getting the agenda together and all the flash docs and everything organized in a way that will flow easily. I'd like to thank our extension partners again. Without their monitor support, our extension program would be a very small version of what we're currently doing. So thank you for joining us today. We are continuing tomorrow morning at 9 a.m. with Khan Kurtrell as our first speaker. Once again, a blend of viticulture and enology. And I hope to see all of you tomorrow. Did I forget anything, Karen?
No, I think you got it all.
So thank you, everybody. Hope you have a great rest of your day and a great evening, and I'll see you tomorrow morning.
See you tomorrow.